like the theme song for that, the very the Pacific Rim movie. Nope. And I have to kind of stay here.
Well, I can give you the radio link.
Well, you're filming though, right? Or is it just the slides? Actually, if I'm going to do the pointer on the screen, it's probably better if I stay here.
Yeah, you look like got the roaming mic.
Okay. No, this will be fine. Oh, I haven't plugged it in yet. That's probably why. It's more open. Feels more, less dungeon-like. Oh, you do? You like it more contrast?
Okay, I'm pretty sure we have a quorum here. Welcome back. Any any administrative, operational, whatever questions that I need to answer for you? Everything's going smoothly? Yeah. Okay. All right. We it's a really long course, and we're like only have 4 days left of it, and it's just this funny feeling right now of it's ending soon, but it's also it's also been a long trip for you guys. So our first speaker today is Vince Calhoun. Vince has a very special and long relationship with this course. He's actually on our external advisory board. He's been back many times because we can't find anybody who can do what he does better, which is, I'm sure you'll see in a moment. Vince actually is trained as an electrical engineer, but even during his undergraduate work, he was already starting to get into biosciences from the appearance of this, and after getting his PhD in electrical engineering, has been active principally in imaging and image analysis. He is now a professor at the University of New Mexico, and he's the director of the Mind Science Institute. I'm trying to remember exactly what the titles are because they're confusing with this. But he is the principal scientist, and he has a very long history of new discovery work, principally in analytic methods like independent components analysis, which is really the feature of the talk we're going to hear today.
Well, thanks. It's a pleasure to be out here again. What I want to do in the first talk here is just go through group ICA and sort of talk about the different choices that you have to make and some of the implications and some of the results, et cetera, what you can do with it. Before I get started, though, I do like to show kind of this one slide just to kind of make a comment about how I think in neurosciences we're still not that good about coming up with good ways to visualize the data. And this is one sort of example of an approach that maybe could be helpful. For example, you see all these pictures of activated brain regions with colored blobs, but we never show kind of what's underneath the threshold. So this is kind of one way to do that. It's not the only way, but for example, if you have an outlined region here, you can still see where the significant regions are, but you see the patterns underneath. And in this particular case, if you look, this is sort of an oddball activation task. You can see underneath these blue regions here sort of outlining kind of a default mode network. Basically, they're going down with the task, and so it's actually informative and useful, and it doesn't waste all that space, that empty space. So if you're interested in doing something like this, we do have MATLAB scripts up online here at this website to enable you to do that kind of visualization. So I'm not going to do as much of that in my talk as I should. I just wanted to kind of mention that as sort of stimulate. Be thinking as you're getting into this about different ways to visualize the data and kind of get underneath the hood more. So here's the topics that I want to go through, and I'll start first with just kind of in the context of seed-based analysis versus ICA, or sort of relating the two of them. You've probably seen lots of maps like this. This is a default mode network. Here, estimated using independent component analysis from REST fMRI data. If we take a seed from the posterior cingulate here and correlate all the other voxels with it and threshold that, we get our seed-based map here. Again, the good news is that though they're not identical, we've consistently seen convergent evidence from different approaches. So, and likewise, if you have a task, auditory oddball task in this case, underneath the task modulations are all of these intrinsic networks that we've been talking about so much in the field in the past several years. Here again is default mode network from ICA, and then if you use a general linear model approach to sort of look at task deactivation associated with a target response, you get that map. So the good news is we get kind of consistent areas, and then each approach has different strengths and weaknesses. Seed-based approaches and ICA approaches are actually very closely related. ICA is really just finding weighted seeds. You can think about it that way. You're basically trying to find a whole map a set of maps that when added together are linearly represent your data. And so once those maps are fixed, once you've found the components, it's a linear model. We'll go into this later in the talk. And you can directly relate them. So again, very similar model. You can do things sort of, you can bring these two approaches together. You can use a hybrid approach where you basically use ICA to find seed regions, and then you can use a seed-based approach on a subset of those regions or all of them. You can do other variations where you give ICA seeds or maps from other studies in order to sort of steer the analysis towards a particular network you're interested in, and that's called spatially constrained ICA. Something to keep in mind as we do this, I call components all the time, I call them networks, okay? So, but it's really important to define what you mean by that word, because there's a lot of different definitions out there. Networks versus, so in this paper cited here, we kind of talk about the different definitions that are used, and there's probably 5 or 6 at least out there. So keeping in mind, so ICA is trying to sort of find a global solution that represents your each component with one time course. Again, I'll get into the details in a minute. So it's sort of a global solution. If we think about what the general linear model, you guys have had the GLM lectures already, right? So if you have a general linear model or even a seed-based approach, you're taking one time course and then you're trying to sort of correlate all the other voxels with that time course. And you've probably heard before, if you have a case where A is correlated with B and A is correlated with C, that doesn't necessarily imply that B is correlated with C. So here's just a little illustration of that. We have on the X and Y axes correlations between A and C and A and B, and we can have them both correlated at about 0.75, and then what's shown in color on the plot is the minimum correlation that's possible theoretically between B and C. And there you can get a minimum correlation of about 0. So you can have a case where you have no correlation between two voxels that pop up in a GLM analysis. So you kind of have to keep in mind, even though we talk about these as networks and interpret them, they're not necessarily directly correlated or even related, other than via their relationship with the time course that you fed it. Okay, so now this is the slide that I show quite a lot, which is sort of thinking about coming at the brain with a very simple model. So here we have elastic scattering data, and say I'm in the shower one day and I realize, hey, this data is probably going to be well fit by a model based on the backs of elephants. Okay, so I go, yes, all right, I get a great fit. I'm going to do a good thing. I'll predict my next data set and write my science paper and be famous. It's a silly example, and I'm not meaning to say we shouldn't be doing modeling very precisely in lots of context, but to complement that, we need to really look at underneath the hood. There's a lot of relationships in the data. It's very complex data, and we're coming at it with extremely simple models that miss a lot. As George Box said, all models are wrong, but some are useful. If you had a perfect model, you would have the thing itself, right? A model's meant to sort of be a simplifying set of rules or descriptions. And my favorite is, I believe in ignorance-based methods, because humans have a lot of ignorance, and we should play to our strong suit. So let's try to do that. Okay, so getting into ICA, it's a type of what's called blind source separation. You may have heard of this cocktail party problem, where we have multiple conversations in a room. We have microphone recordings around the room. And each microphone, if you were to play that recording, could hear all the conversations. And the goal is to given the microphone recordings, estimate the individual conversations and sort of clean them up into their separate pieces. The ICA model, in particular, the one that we use typically for fMRI, linear ICA, is you're assuming the sources are independent, statistically independent. So these conversations are independent of one another, and you're assuming that the mixture is linear. So essentially, the farther away the microphone, the softer the conversation gets. And so given those two assumptions, we have ICA. How many of you have heard of, have you talked about principal component analysis? Is people familiar with that, I'm assuming? So what PCA is doing is trying to take a data set. So here we have, say, microphone 1 versus microphone 2, and you're trying to basically say, let's find components or vectors on that data set such that, in this case, the first one, if I project the data onto it, I capture the most variance possible, maximize the variance capture. And then in the second one, in this case, is trivial. It's orthogonal to it. So it's using variance, which is called a second-order statistic, in order to do that. ICA draws upon higher-order statistics, not just variance. And it's looking for directions that maximize, once you plop these two components down and project the data onto them, it's trying to maximize the independence between those components in some way. So you have to define what independence is and maximize it, amongst those two components. So this sort of straw man general linear model, kind of the simplest we can do almost, is really say we have two conditions off and on. Maybe we have our convolved hemodynamic response function, and you're essentially fitting that to your data, your fMRI data. At each voxel, you have a time course, and you fit the model to the data. You get a goodness of fit, and then using that, you can get a map that you can threshold and say something about these voxels look like the model that I've come to it with. You can write the GLM like this in matrix form, where I have time by voxels. So for the first time point, what I've done is taken my three-dimensional brain, taken all the voxels in it, and flattened them into a long row. And that's the first row of this data matrix. And I build that up. Then the GLM is essentially my design matrix, which I fix ahead of time. So I have to decide what my model is. That's my model. This is time in this direction, and the number of regressors here. And then for each voxel, I fit parameters or weight parameters or beta weights associated with each column of the design matrix. So that's my GLM. ICA, linear ICA, can be written exactly the same way. The difference being we haven't told it what the design matrix is ahead of time. We're figuring it out from the data. And the way we do that is we maximize the independence among the images in these components. So we start from some random guess, and then we work to try to maximize independence among those images. Sort of to give you a little more sort of intuition, suppose I have a video with pumpkins on a step, and so that's a spatial temporal data set. It's 3 dimensions, 2 space and 1 time. And if I do spatial ICA on that, I might get something like this, where I have 4 images, 4 components here. The first one might represent the background, where it's not really changing over time. And then each candle, assuming there's no wind correlating the candles, and they're kind of in their own little world flickering, has its own component. And I can see from this parsimonious summary of this complex video, fairly complex video, that candle 3 went out about halfway through the night. So you kind of see, this is kind of what it's doing, is sort of simplifying, trying to represent the data in a smaller dimensional space that we can visualize. What we typically do for fMRI is with ICA, we still are interested in, if we have a task, doing modeling of that task. And so what we do, though, is instead of doing the modeling up front on the voxels, we push it to the end, and we model the time courses coming out of the ICA and treat them as if they were sort of like voxels in a GLM. And so, for example, in this case, I have a simple task with 2 conditions, 2 stimulus conditions and a sort of implicit rest here. And I model that with 2 regressors, and I have an ICA time course that comes out for each of my components, and I can fit a GLM to that ICA time course. So I'm showing you, this is a screen from the GIF software we'll look at later. The pink time course is the component time course. And in the blue curve here is the fit, the GLM fit to that time course. And then I can get statistical information about the task, the degree to which the task modulates that component by looking at these regressor weights. These are my beta weights here. So component 3, in this case, very heavily loaded upon regressor 1. And component 10 was modulated by the stimulus associated with regressor 2. And I can take these numbers here, and if I have a number of subjects, I can do GLM tests on them. So I can do, 1 sample t-test, for example, to test against the null hypothesis of it not being active. Is there actually a significant modulation of this component with that task? Does that make sense? One thing to keep in mind, and actually a lot of the tools and algorithms that are talked about throughout this course actually have a little secret under the hood, which is that they're stochastic in nature. So you don't get exactly the same solution every time. And so like with ICA, we're starting with a random guess, and then we're optimizing some criteria to get to maximal independence. If I started again with a different random guess, ideally I get the same solution, but in practice you get something that might be a little bit different. And so the way we deal with this in GIFT is we simply run the algorithm multiple times and take sort of do clustering and take the sort of centroid as our answer, which is a stable solution. And there, at least then, if someone else runs it again, they're going to get a solution that's very similar to what you have reported. And so what I'm showing you here are results from 5 different algorithms, Infomax, FastICA, JADE, SymBek, and Amuse. And here is a cluster plot which basically shows for each, this is just one component on the left, and then this is all the components. And you can see that some of them are very tightly clustered. They're kind of all by themselves across the different runs. They don't move around. Some of them here, like 25 and 20, they tend to sort of cross over. They get mixed up between themselves. And that's the kind of information you want to know if you're interpreting the results here, because you want to be careful about those and make sure that you're not reporting something that's not very stable. Okay? Now, group ICA is sort of an extension of ICA in that we're basically trying to do one sort of analysis on a group of subjects and make some inference using ICA. It's not quite as straightforward as the GLM, because in the GLM you have a fixed design matrix. So you can simply just add those up and sort of do them in a group level model. In ICA, 2 things to keep in mind. The components come out in an arbitrary order. When you run it again, you're going to get different component numbers. So the component number doesn't mean anything. 2, the scaling of the components is sort of arbitrary in a sense that we have a map times a time course plus another map times another time course. If I multiply the map by 2 and take the time course times 1/2, my solution representing the data is still the same. So you have to do some things to sort of scale that data. With group ICA, there are lots of flavors of group ICA. The simplest would be, well, let's just run a bunch of single subject ICAs and do that, and then do some sort of clustering or correlation or something to kind of figure out which components match. And if we can find matching components, then we can do tests on the voxels or the time courses for those components. The most widely used one is using what's called temporal concatenation. So you basically take your data matrix here, where we have time again by voxels, and you simply concatenate in the time domain. And you estimate sort of one large group ICA on that full data set. And so you've come at it from the assumption at the beginning that you want to do a group analysis. And you've told, it's basically a model assumption. You basically said, I want a group model. And if you do it that way, you can then take the final sort of average group solution and go back to the original subject data and calculate subject-specific maps and time courses. Okay? And that's called back reconstruction. I'll talk about that in a second. You can also concatenate in space, and you can just sort of average everything together. These give you, not that different group-level solutions, but they're not as optimal for various reasons. For example, in this case, you're kind of assuming that the time courses are synchronized across the subjects when they're not. In this case, you're sort of assuming that the maps, the networks are sort of at least some level consistent. And I'll show you examples where we deviate from that later. And then the one on the far right here is a tensor-based approach where you basically have time, instead of stacking it on a plane, you've built up a cube. And so you have time, voxels, and subjects. In this particular case, you have, for the time course, you're sort of assuming, again, that we have synchronized time courses across the subjects in the time course. And you have one sort of temporal mode that represents the group, and then one map that represents the group for each component. And then for each subject, you also have a weighting factor, a single parameter that sort of characterizes how strong that is. And if Yeah, and so sort of at a high level again, what we do, again, the most widely used approach is temporal concatenation. You do this large ICA, and then we basically have a back reconstruction step where we take either the map that comes out or the time courses associated with a given subject and go back to the original subject's data. And you can think of this as sort of projecting the subject's data into that group space and looking at the maps and time courses for that subject. There are various ways to do this. You can do it using sort of a PCA-based solution. Something I didn't mention is that we use PCA quite often in the processing pipeline for group ICA to sort of compress the data down. PCA is very good at compressing data. And then you do ICA to sort of find the components. And there's usually two levels of PCA, one at the single subject level and one at the group level. And you can sort of invert that process to get to the solution. That's PCA-based. You can do something called spatial temporal regression or dual regression, which is sort of a regression-based back reconstruction. In that case, you're basically trying to invert the linear model. You're sort of doing a spatial regression followed by a temporal regression, treating them as seeds, basically, seed maps. So in the end, what we get out are images and time courses for each subject, and you can do a whole bunch of stuff with that. So for example, for each component, we can do tests, voxel-wise tests, using the GLM framework. So you can use SPM, AFNI, FSL, et cetera, to do voxel-wise tests on the component maps, exactly as if they were sort of contrast images from a GLM, except they're components now. And you can do that for each component. For the time courses, you can, if it's a task, you can fit the time course to the GLM model and ask questions about how much does the task modulate the component or each component, how much does the difference between conditions appear in each component, et cetera. We can look at the spectral information in the time courses. Has someone talked about ALF yet in connectivity or? Okay, good. So amplitude of low frequency fluctuation. So it's similar. We've been doing this in ICA for many years, where you're basically looking at the spectra and you're looking at high to low frequency power, or you're looking simply at all of the spectral bins and sort of testing for them across subjects and groups and things like that. And then you can look at what's called functional network connectivity. Essentially, we have time courses for two components, say, for each subject. And so we go to one subject and we have those two time courses. Instead of just looking at a component map, which is a measure of sort of, I'll call it within network connectivity, right? Everything that's got a high weight in that map is strongly correlated, okay? Same for this other component. And you can also look at the among network or functional network connectivity, the cross-correlation among component time courses. That can be very useful. Indeed, that's one of the most, I sort of recommend that people start there in terms of starting to pull out information from ICA solution. So back reconstruction, I kind of already mentioned, we've got two PCA steps, and for PCA-based, we're simply inverting that solution in order to characterize an individual subject with its map and time course. The spatial temporal regression or dual regression approach is, instead of starting with this time course matrix, you start with this aggregate spatial map, or sort of the average map across all the subjects. And then, so follow me here, what we do is we take we go to the first time point of subject I, and we have, for that time point, we have an image for that time point. And we have, however many component images we've got out of the group ICA. So we can do a spatial multiple regression on the subject's image to get out beta weights associated with each component that was put into that regression. Those beta weights represent the first time point in the component time course. And you would do that for the next image and the next image until you build up the whole time course for that subject. You'll build a time course for every component for that subject. Then you take the time courses you've got and you go to the first voxel of that same subject and you pull out the time course and you do a multiple temporal regression and you get beta weights for each of those, which represents the first voxel of each component for that subject-specific component. And you do it for all the time courses. You need coffee a lot of coffee to get through all of that. But if you want to get into the details, this paper I would highly recommend. It's got all the math in it. It's a very detailed analysis and comparison of a bunch of different things. Just trust me. Just trust me. Okay. And this is just saying basically that all the different things we looked at in that paper, basically, because in the I don't know, it's been since early 2001 or so was the first group ICA paper. So since then, there's been a lot of different flavors out there. So we tried to sort of summarize a bunch of those flavors and look at how they compare. You know, bottom line, there's a lot of you know, a lot of overall similarity, but the devil's in the details. So here we have sort of 4 simulated bits of information. One is sort of the overall component. So there's 8 components here. And then we also simulated subtle differences, which is what we typically are interested in neuroimaging, is not just, oh, look, I got default mode network, but how does that default mode network relate to disease or relate to some variable that I've captured or some information about that subject. And so this is a binary covariate. That would be like a group level covariate. Continuous covariate would be something like age or IQ, and then the interaction between them. So these are more subtle, small changes on top of the main effects. And if you look at we're just comparing here 2 different back reconstruction approaches, a PCA-based and a regression-based approach. And for the main effects, you can see that they look really similar, right? So they get a solution. They're not that different on the end. If you look at the more subtle changes, you do start to see some divergence here. So for example, in this binary covariate here, PCA-based captured it well, but regression-based did not, primarily because this small change isn't well reflected in that average group map that was used as the seed to go in to the whole regression-based approach. And the same goes for the continuous covariate. So here you're getting into very small changes and you're sort of quibbling about where you put that last piece of variance, essentially. But just to be aware of, and there's actually a third kind of back reconstruction. I haven't sort of updated my slides, where instead of going back, you can just keep going. So you do group ICA, you take the group maps, and you can feed those in to what's called a spatially constrained ICA and estimate components from one subject using those maps as priors. And so that gets you a subject-specific map. Yes? Okay. Come on, Mark.
Okay, so does this then mean that for between subject groups with clinical populations, for example, that ICA is going to be less replicable across studies, right? Because I might use a slightly different method, but also clinical groups tend to vary a lot between studies. So for example, one depressed sample is not necessarily equal to another one. Now that's not the fault of ICA, but I guess my question is this sort of going to be consistent, where I might find different models because I used a multiple regression at ICA?
Right. I think there's at least as many, if not more, flavors of the way the GLM is implemented. So this is really more a question of, is your methodological approach documented in a way that someone can replicate it, right? I don't think that is as much an issue with ICA. I think there are some different ways to approach it. You can sort of, you know, use one data set as a reference. You can use both groups as the reference, et cetera. But as long as you do those things consistently, you know, and I'll show some examples of how it handles subject variability later. But yeah.
Sorry,
sorry.
before I give it.
He's got the
give it
microphone,
up.
so he
yeah,
wins.
I got it.
He
right,
wins.
so
Yeah.
Sorry, guys. And so then the other quick question is, so in this case, you would pull the subjects together across group to produce the ICA. And
Well, let
then
me
do
get to some examples
Okay.
later, because I think I'm not quite there yet. So
Oh, okay.
this is really just kind of introducing the approach. But yeah, and then ask me again if I haven't addressed your question there. Okay. My general answer to that is, if you're trying to compare statistically, then you should do it all in the same model. That ideally, and you know, that would be where I would start. Another way to do it would be, I have this huge database and I want to go calculate things from new subjects. Well, you could use that as to generate, there's a whole bunch of websites now you can go to Randy Buckner's site or Mike Grace's site or our site or lots of other sites, the Human Connectome site, and pull networks, components from that site and feed those in to these. And basically that could be your approach, right? So there's a lot of strategies, just like for any analytic approach to these data to handle it, and the devil's in the details. Yes?
Great. This is a clarifying question because I think if I don't get it now, I'm going to be lost later.
Okay.
Can you just explain exactly what the axes are on these matrices? On these
Oh,
matrices?
okay, yeah. So sorry, these are squished, but these are 8 sort of fake components that we made. And this are sort of characterizing the component. This is the mean of the component. This is a subtle binary so it was like we varied across groups, this little region, the blue region there, across groups a little bit for that component. The voxel weights were a little higher or a little lower.
So within an individual box that is surrounded by white.
Each box in that
voxels.
is a component.
And so on the x-axis of a box would be voxels, and on the y-axis is something else?
Oh, so this is an image, voxel by voxel.
Okay.
So it's a
not
fake.
brain,
It's a fake 2D
thing.
image.
Thank
It's
you.
not a brain.
That's helpful. Okay.
At least I don't know. Some brains might look like that, but hopefully
you.
not mine. So sorry, I should have explained that before I started. Yeah. So yeah, this is just this is a way to kind of do a sanity check on the methods. So again, now the brains are circle. The fake brains are circle. Okay. But here we're looking at, well, why not just, everybody always asks, I should do single subject ICA first because I don't want the group to tell me anything about this whole, to influence it or bias it, right? And and those are good questions actually to wrestle through. But one thing that you leverage if your goal is to do group inference or group level studies or just denoise, right? Having access to more data actually can be helpful. And here we have a number of components pulled out of a larger analysis. There's 6 components in the X direction here. We varied, for example, lots of things. So the contrast to noise ratio was made sort of noisier or very small for certain components, and it was large for others. And so here is one subject estimated with a single subject ICA approach for each of those. And here is that subject's representative map from a group ICA analysis. And you can see here that in some cases we have a little bit of mixing that occurred in the single subject analysis, which doesn't appear in the group analysis. And likewise here, you can see even for the low contrast to noise ratio, these 2 components here are a lot noisier than this one because they were designed in the simulation to be that way. And that was well captured here, but you almost completely lost it in the noise here for this single subject analysis. And this much more detailed analysis of that is in this paper here. So I put the references below, so you could look up the papers if you wanted to. So then let's just, simulations are good at sort of showing how great your method is, but they're good. They're really meant to be, does your method do what you think it's doing? A, and B, how easy is it to break your method, right? And so push the limits, right? So here we're going to try to do that. Let's use a simulation where we have so we have a toolbox called SimTB, which is at this website here, which basically kind of lets you. It has a sort of number of example components you can pick to, and you can pick a bunch of different parameters, and it'll mix up a data set for you, and then you can analyze it with any software you want, including GIFT. Anyway, so we did that, and we varied things like, for example, the component sort of shape. So in this case, we have a component that has sort of an outline that looks like this, and what we did is across different subjects, we rotated it a little bit. Okay, so the shape is varying across subjects. Here is the estimated profile for that component. And you can see it does a pretty good job at capturing component variation in the rotation. Here is the ground truth for 6 example subjects and their representative estimated map from group ICA. And again, you can see it captures that rotation quite well. So there can be variation across the subjects in these maps, and it can be well captured. Likewise, if we take a little component and we move it around to different positions, you can see here is the ground truth. Here we do pretty well, but except for the extreme cases, we start to do a little bit worse, and that's where you sort of have things that don't overlap at all, which would, by the way, also not do well in a GLM model because the voxels wouldn't overlap. And so here, but you can see in this case, we do pretty good in estimating the position. It's moving around and we're getting different positions. In the extreme case here, we still get the position, although it's sort of attenuated a little bit. Okay. Amplitude is well estimated by ICA. If we have components with strong activity or weak activity, we can capture that. But speaking to the scaling ambiguity I mentioned before, the scaling issue, if we look just at the time course or just at the spatial map, we don't get a tight curve because remember the scaling is sort of distributed across the time course and the map, right? You can move it around, multiply this by 2, multiply this by 1/2, etc. And so you have to lock that down somehow, or calibrate it or scale it. And there's various ways to do that. And if you take the joint metric of both time and space here, we have a nice tight correlation with the ground truth in the scaling. Okay. And then, since we were interested in looking at cross-correlation among components, that's what this is looking at. How well can we capture it? And it captures it really well. This is showing 2 component distributions where we have a high correlation between components and one where the correlation is nearly 0 between the 2. And again, it's pretty tight on the line. And that's all within that paper there that's cited. I'm going to show you some examples from data sets. I sort of always throw in one slide here just to mention there's a lot of thrust in the field now to do more sharing. We need to share more. We need to get along, give people data, And so we have a tool called Coins, which where we've got some data sets shared, and there's a lot of other data sets that will hopefully soon be shared. And there are lots of tools out there to do this, XNAT and others. And so it's a good time to be getting into neuroimaging because there actually are more and more data sets now, many more than were available. So you could, if you're someone who likes to do analysis, you could analyze pretty much forever on just the data we already have without collecting data. Although I'd recommend you collect at least a little data to kind of learn how the data works. So anyway, you can see here the distribution of sort of people that provide data and people that use the data, and there's actually quite a demand out there. So this is an example data set of healthy controls, 600 healthy controls that did a 5-minute rest fMRI experiment, all on the same scanner at the Mind Research Network. And I'm just showing you sort of a high-level group ICA analysis, right? So what we get out is each of these 3 orthogonal views is one component. And so I have a bunch of components shown here, and they're grouped based on their anatomic information. So for example, visual is grouped here, default mode grouped here, etc., sensory motor, okay? And so this is the within network connectivity. If you want these maps, by the way, they're at this website here. And then this is a matrix that sort of represents the cross-correlation among the component time courses. So each subject's time courses were cross-correlated, and then you can average those across subjects. And you can see a lot of useful information when you look at this kind of thing, right? So for example, the visual components are all, this kind of blocked structure in the matrix you can see. I've organized them intentionally using these categories over here, right? And you can see that the visual components have high red values. They tend to be correlated with themselves, right? You can think of that as like within domain, you've got strong correlations. Here's default mode here, correlated with itself, but weakly anti-correlated with these other networks. Again, consistent with what is seen quite often with default mode. And then here's a spectral information. One thing we do a lot with the spectra is we use it to tell us whether something about artifacts. Are these good or bad components? This question will probably come up. How do you pick the components that are good and bad? We use this sort of high to low frequency power ratio as a guide for that. And this is an example here where 4 or 5 different sort of ICA experts manually went through all the components and decided what was good, what was bad, what was kind of in between. And they coded them yellow, red, or yellow, red, or green. And in this plot, I'm showing basically the high to low frequency ratio versus the dynamic range, which would be this point to that point. And you can see all the greens are up here in the far right corner, and all the reds are in the lower left corner. So the more high frequency power you've got, the more likely a component is to be an artifact. And that makes a lot of sense because, for example, motion, right? There's no hemodynamic filter on motion. It's just a rapid movement or so. So it's going to have high frequencies in it, more so than a bold response. Okay? And that paper there kind of goes through the details. At a high level, we can look, for example, then at statistical tests for all these components and time courses. So this is the voxel-wise relationship with age. You'll notice, and I'm summarizing it sort of by each component with sort of, one average value. And they're all sort of pretty much negative. So as people get older, their within-network connectivity tends to go down, okay? With the exception of, for example, basal ganglia here, which maybe that's the wisdom region of the brain. I don't know. And then here we have gender effects, females versus males. Generally, females had a little bit more within-network connectivity than males did in a few of the components. And then we can look at the FNC, sort of across. I think you've heard a lot probably about, rapid imaging and using the multiband sequence from the Connectome project, et cetera. And one of the nice things about that is again, if you look at the spectral information, you can start to sort of differentiate the cardiac signal more cleanly because it's not aliased. If you have a TR sort of below, say, 400 milliseconds or so, you're getting into a range where you can characterize cardiac. And so this is just one example of a group ICA component whole brain analysis where we have this power range here right around 1 hertz. Okay. Getting to how many components do we pick? 1, 10, 50, 100, 200. There is no right answer there, sorry to say, but you can use several techniques to kind of estimate that number, okay? And those work. They tend, though, to preferentially try to minimize that, keep that number small, so that you're sort of balancing number of parameters against how much you fit the data, how well you fit the data. But more recently, there's been a trend to kind of go higher than that, up to 100 or 200, because again, think about this as a spectra. The more components you have, the more you're splitting things up. The fewer components you have, the more you're kind of clumping things together. And generally, people are interested in things like, how do these nodes within default mode interact with one another? And you can answer that question if you go up to a higher model order, because they get broken into different components, and then you can model the time course interactions. But that FNC matrix is really useful, because if you keep that information, you can see, as I mentioned earlier, the correlation within the visual domain was high, right? So you can kind of see from that the evidence that tells you whether things would sort of clump together if you went lower in the model order. And you can actually reconstruct a lower model order decomposition from a higher model order decomposition just by linearly combining multiple components at the high level. So you can kind of get both if you do the high decomposition. The other trade-off, of course, is if you do high model order, you've got more things to test, so you have to do more rigid correction for multiple comparisons. Unless you want to do some combining, for example. Okay. All right. I think I'm going to have to go faster here. So just a couple comments on processing issues. Motion is a big issue in the field. ICA is more robust to motion than is a seed-based study. Primarily because in a seed-based study, you're just doing univariate correlations. You're just isolated. You're doing sort of massive cross correlations. In ICA, you're working with the distribution of the whole data set, so it's kind of benefiting from that. Some components get separated out as noise, but that's true. Here's an example of 2 components. This looks like a component that's due to motion, right? Like the brain was tilted a little bit in the front, and so one's negative, one's positive, and it's on the rim here. And if we regress out motion from the voxels prior to doing ICA, you still get that component, right? So ICA is still finding stuff in there that looks like it's motion related, even if you do the best regressions that you can do. However, that doesn't mean we shouldn't be doing those regressions with motion parameters. If you look at the FNC matrix here, and just kind of look at the pattern of the matrix, we had a couple hundred subjects, and we sort of split them into essentially non-movers, your perfect subject, right? Folks who kind of sit still the whole time in the magnet, and then folks that have sort of intermittent spikes in their movement, and others who sort of kind of meander all around. Notice how similar these look, whether we do sort of motion regression or data scrubbing before ICA, or if we do it after ICA. So you take the time courses out before in your FNC matrix, and you do regression after the ICA, then calculate your matrix, okay? Those two scenarios look really close, with the one caveat that severe spikes can influence the ICA decomposition a little bit. And so if you look at the Jonathan Powers measure of sort of, how connected are components that have regions that are far apart versus close together, you'll see this relationship of, the long distance correlations start to go up as motion goes up. And you can get rid of that if you do despiking prior to ICA, and you can do motion regression either before or after ICA. If you do motion regression before ICA, the downside is you have to redo the whole ICA if you change anything about your regression, and B, it does decrease a little bit the regional homogeneity within each component. It kind of disrupts the voxel-wise smoothness a little bit. Autocorrelation, another thing to mention, it's been largely ignored in resting data. So if you have a little bit of smoothness in your time courses, a little memory, time point 1 remembers, is influenced by the previous time point. And there are models in the GLM to do correction for this sort of thing. And we were concerned about whether it would have an impact on rest. And if indeed you look at, the matrix, the FNC matrix, in an uncorrected, this is just correlation. It's well known that autocorrelation influences or biases cross-correlation among time courses. And you can see here, if I correct, if I use a sort of autoregression model to correct it, these look different, right? And so this could be a problem. But we kind of got lucky, because in the field, most of the time, we're doing group studies, and we're doing actually statistical. We're working at the p-value, so we're doing like a ratio of the correlation to the mean correlation or the mean z-value to its standard deviation or standard error, and the correlation is biased upward, but so is the standard error, and so they sort of cancel each other out. And so you can see here the p-values look pretty similar. I think there's more work that needs to be done here. We're also looking at autocorrelation as a value of interest in and of itself. We're calling it autoconnectivity. It's like the voxel being connected to itself, so to speak. And you can see group differences in those parameters. So, yes, Mark?
Yeah, I guess maybe it's what you just talked about in the last comment, but it seems to me that if, in fact, we're looking at an intrinsic blood flow signal, we expect some level of autocorrelation, and perhaps one ought to think about pre-filtering the data to look for that specifically as a denoising step. That is stuff that fills any model that you have on the data. Is that, if it's a reasonable model, ought to help your sensitivity.
Yeah, but you also get this a pre-processing step used in pretty much all seed-based approaches, and is bandpass filtering, right? And so you're inducing autocorrelation on your data, and so if you don't account for it, you're going to have a bias in your correlation there too. So your point is well taken as well, which is that the signal of interest is coming from a hemodynamic effect, and so we know something about the characteristics of that and can use that in the modeling. But getting it sort of low and high, and people are looking at higher frequency things in hemodynamic signals these days. And so once we get rid of all the motion components, then if we look at what's left, we start to see things like, if we're looking at here, healthy controls versus schizophrenia and bipolar disorder. Here's an average spectra, and you see that healthy controls have more low-frequency power. Patients have more high-frequency power, and that's after accounting for motion, okay? So I don't think that's a motion effect. I think it might, we sort of speculated, in some early work about how it might be due to efficiency of communication, sort of translated through the connectivity in the brain. So you have more frequent fluctuations, but maybe a less efficient communication among those brain regions. I don't think it's clear yet, though, what it is and what it means, but there are lots of studies out there, and this is very closely related to all the ALF work that's out there, right? Looking again at sort of low frequency power changes here. This kind of thing would be picked up as an ALF change as well. Denoising, so there are, again, aggressive denoisers and conservative denoisers, and we should sort of figure out where you guys fall in that spectra. But the aggressive denoisers will throw a lot of data away or do very, very hard things. Like, for example, in the ICA context, you can take the approach of, I want to do single subject ICA on every subject. So if I have 1,000 subjects, I have to do 1,000 separate ICAs, pick out the components that are bad, or use some tool. There are various tools in GIFT and Melodic to do this, to sort of try to semi-automatically figure out which ones are good and which ones are bad. Remove them, put the data back together, and then do a group ICA on it, right? Another way to do it is to reverse that. You can say, there's some error associated with that single subject process, and each individual ICA is using less data. Let's do it at the group level. We do group ICA, and then you can use the group components and do spatially constrained ICA on each individual subject, and basically let that subject tell you what's unique about it, incorporating the higher order ICA information for that subject. And that approach actually performs as well, or in a lot of cases where errors are made, better than doing the massive single subject ICA followed by group ICA. You can do group ICA followed by single subject ICA, but you don't have to pick the components in that second scenario because the priors basically order the components for you. Okay? Yeah. You can't ask a question. You're in my lab.
Okay. So when we do spatially constrained ICA, where do we put the spatial constraint in the ICA process?
You can give it a whole bunch of maps, a continuous map.
So
So the component image
the W gets estimated
gets
based
weights
on
associated with it. So, okay, and couple other things. Task versus rest. So again, everybody's jumping on the rest bandwagon. There's a lot of cool stuff you can do with rest. The psychiatric community likes it in many ways because patients can rest. Okay, they can't do a lot of tasks, so they have performance confounds with the task. So, and you can get really big data sets that are rest data, right? But I'm sort of advocating here, and I think that's great. But then we should continue to focus on task as well. So here's same subjects that had rest and an auditory oddball task, and the components that come out for those done separately are very similar, right, to one another. But this is an oddball task, and so let's just look, say, here at temporal lobe, and you'll notice, though they're similar, they're not identical, right? And if we do a direct subtraction, you start to see which regions are sort of getting more correlated, or the regions are shifting a little bit in terms of you're recruiting more, different parts of your brain to engage in the task. And even more interestingly, this is schizophrenia patients and this is healthy controls. And you see that the change between rest and task interacts with the diagnosis. Okay, so the patients, show different changes between rest and task than controls do. Okay, and this is what we look at all the time in task data, right? We're looking at some stimulus versus implicit baseline, but this is extended rest connectivity versus a task-based connectivity. And so I would argue that there's a lot of interesting information there that we shouldn't get rid of or ignore. It varies. I think, yeah, the question was, do you always acquire resting state prior to task? And so, yeah, you're getting at sort of how do you design your study and how do you worry about order effects and things like that. So if I have a study that has disgusting stimuli in it, right? The rest of the study, I'm going to be thinking about how gross that picture was, right? And so my connectivity might be a little different. But if you always do rest first and task later, you've also got, they're more tired, they're in the scanner longer, so you've got issues there in terms of they might move more or whatever. So it's a challenging question. I think you ultimately should randomize things, but yeah. Which this one? In this?
I know it's really hard because you're
Yeah.
right in front.
Should I should not answer them if they don't use the microphone. Okay. I didn't hear that at all.
Yeah, I was, I mean, in this, sorry, in this paper, in this study.
When you say this, are you talking about this slide or what?
Yeah, the slide.
Yeah, okay.
Yeah.
Oh, so for this one, I believe the rest was done first, but I would have to look that up. I don't remember.
Okay,
So.
thanks.
I'm trying to think of individual variability and what you're throwing out doing either one or the other method. So if you start with a group ICA and spatially constrain the ICs that you find in each subject with that, are you throwing out more of the intersubject variability as you would if you start from doing ICA in each subject?
So that's kind of what I was sort of addressing with those simulation slides, because if we do really well on those simulated results, that means we've captured the individual variability. And if you capture it, you're done. You've got the solution. But as you will see, it's not a golden hammer, right? Because as things get further and further away, you can start to see things break down a little bit.
Right. So you can capture things that are kind of in the same
Unique.
spot.
Yes. Well, even if not, right? Remember the rotating one, right? Those didn't overlap at all, and it still did a good job. And we also find, good results, as I think we showed in our 2001 paper, right? So we had very different looking things, right? And they still can get well captured in this model. I wouldn't say that will always be the case, right? But it's remarkably good at capturing, because it's fitting your data, and the back reconstruction approaches work pretty well. So, and so this is again sort of, I'm not going to go into the detail. Maybe I'll skip over this, but this is basically saying, we can look at things like a hierarchy of tasks, right? So we've got rest, a low-level auditory stimulus, audio-visual stimulus, et cetera. And we can look at whether patients are showing, schizophrenia patients, for example, are sort of parallel to the controls but just reduced, or whether they cross, and things like that. And there's a lot of interesting things that you can do there. All the stuff that we already do with tasks, but you can incorporate rest and the concepts of connectivity into this kind of analysis. Okay? And let me just skip over a few things here. So this is a little more, this is just an example of a Sternberg working memory task where we had 3 levels of difficulty. They're shown some words, and they have to respond later whether they've seen those words before, and we can ask them to remember 1, 3, or 5 words. So it's kind of 3 difficulty levels. Okay? So here's my design matrix. I have sort of 3 periods here where I've presented words, and 3 periods where they're seeing responses and recognizing or not those words, et cetera. And so if I do a group ICA on this data, and then I do a GLM fit using this design matrix to each component time course, I can come to this with a question. I can say, well, I'm interested in, for example, group effects or difficulty effects, load effects, or their interactions. So I could just do an analysis of variance, group by load, look for group, load, or group by load interactions. And in this case, 3 components come out that have one of those significant effects. And so this is one that comes out which has got a group effect and a load effect, essentially no interaction. This is kind of a map of some key working memory regions. And you'll see here, I'm showing you the average beta weights for the different difficulty levels for healthy controls and schizophrenia patients. And you see the patients are lower on all 3, but both groups are going up in their response to the task the more difficult the task is. Okay? Yeah.
It just totally left my head. I'll think of it later.
Okay. That was an easy one, yes. Okay. Component 2 is another one to illustrate the point here. This one has sort of essentially almost a group effect, almost a load effect. But I want to point out here, these are negative, right? And this voxel here is blue. So in that voxel, is the task is the task increasing the component time course or decreasing the component? Is it correlate with the task or anti-correlate with the task in that voxel that's blue? But remember, the task beta weights here are negative. Huh? Correlated. So negative and a negative is a positive. Key thing to remember with ICA, you don't know the design matrix, right? We don't know that mixing matrix. We don't know what it is. In the GLM, we implicitly don't look at our design matrix because we already know what it is, and we just look at the map. With ICA, you have to look at both pieces of information to interpret it. And another interesting thing to make here is, so you see that healthy controls sort of go up with the task. Schizophrenia patients go up and kind of have a little bit of a quadratic effect. They sort of taper off at the most difficult. But this negative this voxel here is showing patients are greater than controls. So we'll call that hyperactive in the patients, right? In this one, patients are lower than controls, so it's hypoactive in the patient group. And there's been a whole bunch of argument over patients hyperactivate or hypoactivate dorsolateral prefrontal cortex, right? Well, if we look right here at DLPFC, and I flip between the two slides, they overlap, don't they? Right? So the answer is it kind of depends, right? It depends on where you are in DLPFC, and in some cases we've got voxels that have a mixture of two things, something that's going up and something that's going down with slightly different patterns. So this kind of thing is really useful. The fact that the components overlap with one another is actually very useful because you can separate out artifacts with that. You can also separate task conditions that overlap. Yeah.
Okay, I thought of the question. So when you have sort of the time series of the task itself, right? Can you just talk for a minute about including that in the ICA versus picking out components afterwards and then correlating them with the time series? Like, would the first one be like a PLS instead?
Yes, I didn't show you anything on that, but you're asking me that, right? Is that?
That, yes.
Okay, yes. Yeah, so you can constrain your analysis by giving it the time course up front.
Sure.
And you can do that with ICA too. It's called semi-blind ICA.
Okay.
It's basically, again, on the spectrum of how strong are your assumptions, right? You're making stronger assumptions if you sort of force the design matrix on the data up front but not as strong as if you're doing it sort of univariate GLM way, right? It's
Sure.
still looking at the correlation structure. And whereas in this case you've said, let the data fall where it may, and then let's look at what it looks like. So it's kind of there's no right answer there. This is a stronger assumption, this is a weaker assumption.
Thank you.
Okay. More prior information, less prior information. So, okay. And then this last one here is a nice temporal lobe component, but it's essentially showing a big group effect, but really no load effect. I mean, you can see a little bit here in controls, but nothing is significant. And so this variance here, which is, the spatial map is in regions that we think are related to, sort of affected by schizophrenia, but it's not showing much task effect, so it's going to fall into your residual error if you do a GLM, right? Whereas it gets picked up here with ICA and pulled apart from everything else. Okay. All right, let me just buzz through. I have till, what time do I have to start? I know I've already gotten until 9:30, Mark. Do I have 5 more minutes, or should I? That's my second talk too, though, yes. So, okay, so I can eat into he's saying I can trade off against my 2 talks. Yes. Let me just skip over some of this here. I just want to mention here that you can use, I really like using ICA to do classification, basically to calculate features from the data that you can then feed into classification algorithm. For example, here, if we give ICA very little data, say just 49 time points versus 167 time points, and I look at 2 components, temporal lobe and default mode, as I get more and more data, they're pretty stable, right? So I've got enough information to get a good stable component with very little data, because I've used all the voxels to kind of estimate that. Whereas with the GLM, you can see things start looking really bad. Then for the oddball task in particular, we start to get our temporal lobe, then it goes back down and comes back up. So it's a lot less stable. And so there's a lot of papers out there where we've looked at classification using the spatial maps, or the time courses, or the cross-correlation FNC matrices. This is a more recent one where we basically said we're gonna use this normative data set to do our group ICA, then we're gonna fix the components and use the spatial temporal regression approach to go to a new data set of schizophrenia healthy control, calculate correlation matrices for each subject. So again, the group information didn't tell the ICA anything right there. And then we threw those up on this website, competition website called Kaggle. And the challenge was, diagnose or predict schizophrenia versus healthy control, right? And remarkably, I was surprised. We got over 2,200 submissions to this competition. Each dot on here is a submission. So, these poor guys were doing pretty bad, right? They're less than chance, 30%, right? You just flip your labels, right? So, here we've got, but if you look at the cloud of data here, by and large, it's above 80%, right? The mean is above 80%, and some of them are upwards of 90%, right? So there is a lot of information in this data about, predicting, independent of which algorithm you use. There's a whole bunch of different classification algorithms that are used here. And I think, part of the reason we got such a good response here was probably because people didn't have to know anything about brain, right? We just give them a matrix and for each subject, and then they have to classify. What they do is they get a data set to download and develop their algorithm, and then they get a public leaderboard data set where they basically make guesses of which group they are in between 0 and 1, right? And then you get this leaderboard that comes up, and you see who's beating you and who you're losing, and you can every day up through the end of the competition, you can resubmit your guess, right? And there are tricks to kind of make sure people can't figure out all the labels on that. And so that was really fun. And then the final solution is the winner is determined by this private leaderboard, which is using data that they don't see at all. So if you overfit your public leaderboard, you're going to actually probably do worse. But it was fun watching. This team was like, what are they? Was sort of they're like trying to say they're trying to trick us. They're doing this and they're doing that, and then we need to do this. And there's all these discussions going online, which was really, really cool, actually. So I think we need more of this stuff. I'm going to skip over this and just jump to, I just want to mention dynamic connectivity before I stop, which is a very popular topic these days. Instead of cross-correlating the whole time course here, yes, Mark? Oh.
I have a question.
Yes.
Since we seem to be asking questions along the way.
The what? Say again?
The 88T200 data set, which had a similar competition,
Yep.
and they got around 60% accuracy. I was wondering, do you think that's because of methodological differences, or just something about the disorders, or I don't know if you're familiar with that?
Well,
competition?
I think, technically the winner of that competition didn't use imaging data, right? So there was actually, the groups weren't, there was some matching issues, right? So the phenotypic information was enough to be able to diagnose without the imaging. That was also multi-site. This is one site. The data was all collected at one site here. So but there's a number of differences, but yeah. So instead of correlating the whole time course, maybe things are happening like we have a strong coupling between two components or two regions at the beginning and a weak coupling at the end, right? So there's these dynamic things that we can do to sort of look at changes in correlation over time. And you can do this in the context of group ICA, we'll call it dynamic FNC, where instead of estimating the FNC matrix from the whole time course for each subject, we use a tapered window, for example. And we can estimate the precision matrix from that smaller set of data and do it and shift it by 1 TR and do it again and do it again. So now you get a time series of changes in the cross-correlation among these component networks. So what do we do with that? Okay, now we've just blown up our data, right? We've got a bunch more matrices to deal with. Well, we can do clustering, for example. We can take this and we can cluster it down into sort of 5 stable recurring patterns of connectivity. And this lets us do things like go to an individual subject and say, what state were they in at which point in time. So this subject went from state 3, this highly sort of anti-correlated state, to state 2, to state 4, 5, etc. Right? And there's a lot of cool things you can do with this in terms of summarizing, calculating metrics from it, etc. But one thing that the dirty little secret about all the rest data we have is people actually rest when you tell them to rest. Who would have known, right? Who would have known? And so this is some data from Enzo Tagliazucci where they had concurrent EEG data and they did sleep staging on subjects in the scanner. And so they have these different sleep stages. And so if we just look at awake versus the deep sleep, this deeper sort of near sleep, N3, and so the awake maps nicely onto one of the states, and the deepest sleep maps nicely onto another of the states, and the other ones presumably are doing other things. And he actually went back and he developed a classifier based on fMRI data informed by these EEG sleep stages, got fMRI patterns that matched those, and went to the FC1000 data and showed that pretty much all the subjects in there were falling asleep, or at least getting very drowsy looking by at least halfway through the experiment, and some were were pretty much out the whole time, really tired overnight, sleep-deprived people. So, and here you can see, if we look at one of the states, we get a very nice mapping of the sleep stage measure from EEG, the hypnogram, with this dynamic state vector here. Okay. But you can also do stuff with looking at patients, and again, I'm just going to sort of skip over this, but just to say, for example, here we have 5 states. What we see is that the controls spend much more of their time in these states 1 and 2, these sort of strongly modularized states, whereas these less connected states 4 and 5, the patients spend more of their time. So controls in states 1 and 2, patients in state 4 and 5, significantly different in terms of their dwell time, how much time they spend there. I'm going to skip over this due to time, but there's more cool stuff we can do. And you can also do things that, instead of doing windowing, you could look at the full time frequency information. So this is building on work that Katie Chang did, but you can basically estimate for each state the strength of the correlation, the frequency distribution, and the phase, so the delay. And those 3 pieces of information actually help you separate states from other states. And we show that in let's see, this paper here, which is online at this point here, Yasubi, that you can separate out states that have very different spectral power, but similar correlation strength. And one of them shows a group difference and one does not. So you're sort of able to separate out things you can't separate out if you do this time frequency or coherence-based analysis. Okay? And spatial maps vary in time too. Okay? So lots of tools out there. We'll go over GIFT later. I'll give you some demo. We'll sort of demo that. But probably the main ones that are used these days are the Melodic tool, GIFT, and Brain Voyager. Those are probably the 3 most widely used, and then these other ones are also used as well. But okay, and they can be you can do group ICA these days for at least the Melodic and GIFT software on as many subjects as you want. So the memory scaling issues have been solved for that scenario. So you could do 1,000 subjects or 10,000 subjects or 100,000 subjects if you have enough disk space somewhere to store your data. So, okay, I think I should stop there. I don't know if we're going to take a short break and then we'll come back and for something completely different. We'll do data fusion with largely ICA based.
So a break isn't on the schedule, so it's up to you.
Oh, okay, good. So we're going to keep going?
It's up to you.
Let's keep going. Why not?
Sorry, one question.
Yeah, go ahead.
For the group ICA, given that in ICA we find independent components, if we have an outlier subject, could that subject sort of inject more independent components that is not present in the group, more than like what we get in GLM, for example? So does that inflate the data more?
I think a bad subject would hurt both models, right? But generally, it kind of gets at how many components should you pick as well. What we have found, it's another reason why we like to go higher rather than lower. We usually try to pick the maximum component number based on estimates from the individual subjects. Or, and what you typically get is if a subject doesn't have those components in there, their maps look kind of like noise.
Okay.
And it shows up in the subject that has that component.
So sort of you find the components and then you reconstruct and see which one is noisier. In that way, you find the one that is outlier, so you will not.
Well, you can see it. You can see individual subject maps out of a group ICA,
Right.
and typically for subjects that are, that have something different going on, you can see it in the data.
Okay,
So,
thank you.
Yeah.
How many other people in the room are cold? Okay, let me see if I can do something about the temperature.
I'm fine. I got my ice water up here.
Hi. So I had a question similar to what was asked before about semi-blind or semi-supervised ICA versus the blind ICA. In case if you have, I mean, it works well for, let's say, default mode resting state, but if you have like a really, really busy rapid event design, would it help to then try out something like a semi-blind ICA and, have you guys looked into that and compared the performance of just a blind versus semi-supervised?
We did, so again, the semi-blind approach can be applied in time or in space, okay? I think you're talking about time, right? So that's on the mixing matrix, which is a little more challenging of a problem because you're optimizing in two domains jointly. We have looked at that in not a very rapid design, but sort of a fairly multi-second, like 5-6 seconds on average auditory oddball task, right? And we were able to basically, sort of focus the ICA to sort of pull out a task-related component from that. I don't know that it really matters whether it's rapid or not. If it's really a question of how much signal do you have in there. The group ICA does well at sort of finding task-related variants, but it may be distributed across multiple components, right? So if you want to try to force it to put as much as possible into one, that might be one solution. But as you get into this, if you do this, you'll start to see that the more you constrain, the more other things that can come up sometimes. So you have to be a little bit careful about it. But yeah, I mean it's a I don't know what the right answer would be there, but you can certainly do that. So.
And another question about what's your take on the optimal time needed to get resting state data? We generally at our lab use 6 minutes. We also have clinical populations, so we can't recruit like a lot of subjects, but what's your take on it? And then, everybody wants to do some fun task and then get resting state data as well. So
yeah.
Is there a power issue?
We've looked at, things like 3, 4, 5, 10, 15 or whatever. It kind of depends on what you're looking at, right? If you're looking at really low frequency stuff, then you might want to have it a little longer, but we've gotten great results, even as down as low as 4 minutes. You're still going to be able to pick up a lot of what's going on. So 4 to 5 minutes is sufficient, especially if you're doing multiband and getting more data per unit time. So it sort of trades off also against how many subjects you have, right? Because you can sort of compensate for lower SNR within one subject by getting multiple subjects, right? So there's some trade-offs there.
Thank you.
Quick question. I didn't completely get how the number of components is determined. Is it data-driven? You said that you can choose it to
We'll
some
do
extent.
both. We'll do it. I'll show you how to do it in the GIF software later in the demo.
All right.
But a lot of people just pick it. I like to flip coins.
Do you have? Can you comment on like the algorithms that perform best for the schizophrenia classification, or is that?
Oh, I have a slide I can show you later. I don't have it in the talk, but
Okay.
there actually is a paper referenced in underneath that slide from a machine learning conference, and it's got a lot more detail in it and it tells you what the 3 winners were. And we also did this weighted voting. So we took multiple performance scores, and we just took, like, say you take the top 10, and if 6 out of 10 vote patient, then it's patient, et cetera. And we were able to beat the best score all the way up through 60 of those, right? So there's a lot of information, that we're missing in each individual approach. So. Boy, once you remember that question, you just keep coming back. Yeah, so.
Sorry, this is like a thing that we've been talking about a lot about in our lab, so I'm really interested.
Sure.
Okay, so my question now is, so in my lab we look at prefrontal amygdala connectivity a lot, and one of the things that we find is that if you look at a resting state connectivity, it's often positive, but within a task we often look at like emotional images, right? And we see a negative connectivity if you do a PPI. So one thing we were wondering, and we've been talking about, is this idea of like, do we see positive connectivity over a long period of time? Like it's a low frequency connectivity, and then within a PPI, you're just seeing a very like transient negative connectivity in a short period of time? Or like, and so we've been talking about maybe doing like an ICA on task data, but I didn't even know that it was being done. I guess I haven't looked that much into the literature. So can you speak to that at all?
Yeah, I think you should.
Okay.
I think you should. It really is informative about task covariation. And there's hundreds of papers out there where people have used group ICA on task data. So there's a lot out there to look at. So it works. It does a good job, and I think it'll be complementary. It'll kind of give you another piece of information that you can use in figuring out what's going on. So.
Great. Okay, thank you.
Yeah. Okay. So let's see if I can get through this. Do I have till 10:30 or 10:45? I thought I had till 10:45, and then a 15-minute break, and then 11.
Well, I think our break is at 10:30 until 10:45.
Oh, okay. Well, I'm going to be.
I'm sorry.
skipping some
What does it say on the other thing? 10:45. You got 10:45.
Yes. Okay. All right. Good. I'm still going to be tight, but that'll help a little bit. So now, you've seen all these different beautiful kinds of data that you can get from the brain with MRI, EEG, MEG, et cetera. So, and genetics, should we try to be combining this data together somehow, right? So multimodal. And I'm going to go through sort of multivariate approaches, largely based on either ICA or CCA, which is canonical correlation analysis, and talk about kind of how that's been done in the past, and what it can tell us. So we have a lot of complementary information. No acquisition approach to date is going to be perfect, and we're so far removed from what's really going on at the very neural level and cellular level with live human imaging. I think we need these complementary pieces of information to kind of help us. So this is my son. He's now 12, but in this picture he's doing data fusion, right? So we want to try to maybe better than this. We want to try to leverage the complementary information. So for example, EEG is the natural one to talk about. You have high time resolution, but it's coming out from electrodes and very low resolution space. fMRI, you get localized hemodynamic activity, but the timing information is a little bit sluggish and harder to talk about. And then we have lots of MRI-based modalities. Just to put sort of a scaffolding down on this, you can think about doing a joint analysis. Say you had two modalities. We could do, say we had an fMRI activation map from a task, and we have a gray matter map. And you can do cross-correlation at all of those voxels across subjects, right? And see where structure and function from your task correlate. So that's straightforward. It's hard to visualize, because you've got all the voxels to deal with. You can do region-based, where you'd say, I'm interested in anterior cingulate and dorsolateral prefrontal cortex, and these regions are these atlas-based regions, average within, and then do cross-correlations or something across modalities. In here, you things like structural equation modeling, et cetera. dynamic causal modeling, where you're sort of looking at, that would be sort of, that can be done within an fMRI experiment when you're going across regions and looking at time and sort of dynamic information, but you can also do it across subjects. And then more transformation-based, which is sort of the data-driven approach. So SVD, partial least squares, CCA, and ICA, et cetera. And that's what I'm going to be talking mostly about today. And so, why do we want to do this? Well, for example, in this case, we have two clouds of data. Here's modality 1, here's modality 2. Sorry, two groups of subjects, patients and controls, say, right? And the marginal distribution from one modality or the other modality, you can't really see patients and controls in that data set. But if I were to draw a line here and plot that distribution, I can start to see the distribution separate, right? So it's really about finding this joint information that we can't see with one modality or the other modality separately, but together we can see, hopefully. Okay. Another, I've already talked about sort of the robustness of the approaches that use multiple voxels, like ICA, for example. So I'll just kind of jump over that to save time. The other thing that I'm going to do in a lot of the examples I give you here is what's called a feature-based data fusion. And so what I mean by that is I've tried to reduce the data down in some sensible way, right? Because we're already struggling with one data set. How complex is fMRI, right? How complex is brain structure or diffusion tensor imaging? And so let's try to reduce that down a little bit to just ask some initial questions. So for example, in fMRI, maybe I have a task-based map that I can calculate. Or I want to look at default mode, for example, or an ALF map. And for structural MRI, maybe I compute a gray matter map. So then I have a single variable per subject with multiple observations, right? So now I've gotten rid of the time from fMRI in that case. So that's what I'm calling feature-based analysis. And we're probably going to lose a little bit of information in there, but we're going to make the modeling much more tractable, and then we can back off of that later. Okay. Feature-based ICA, actually, if in fMRI data, if I give ICA contrast maps from a GLM, say from a sensory motor tapping task, right? What's that going to do, right? Well, in this case, what I'm showing you here on the left and on the right is a group ICA of the networks that came out from a group ICA of a large data set, or an ICA that was done on a sort of subjects by voxels matrix, where the voxel map here is a GLM contrast map from a motor tapping task. Okay? And look at how similar those, you can find the networks, because the connectivity information is actually biasing our GLM map, right? So the variance across subjects in the GLM maps actually contains information that looks remarkably similar to all these intrinsic networks that everybody finds. In an extreme case, you can go to, this is a paper from Steve Smith, where you can go to a meta-analytic database and just pick regions out of papers, put a little Gaussian blur on that, create a map, do that for multiple thousands of subjects, do ICA on that, right? You wouldn't think there'd be a whole lot of information in there other than kind of the coordinates, right? And you get these network-like things that come out of it. So, they're everywhere. So this is too dense here of a slide. I'm just using it, giving you the reference to the paper. But there are a lot of approaches out there to do multivariate data fusion, okay? I'm going to touch on some of these examples here. So we have MCCA, joint ICA, combination, and then there's things like parallel ICA, for example. Okay. I'm going to skip over that and just give you sort of an example of joint ICA. So the first model we'll talk about is joint ICA. So in that model, what you're doing is you're basically taking a feature like a contrast map from one subject and another feature from that subject, like a gray matter map, or let's in this case focus on two different tasks. We have an oddball task and a Sternberg working memory task, and we look at the target-related contrast map and the recognition map from the working memory task. So we have 2 images per subject, right, for a bunch of subjects. And what we're doing in joint ICA is you're essentially analyzing them side by side, trying to find components that are maximally independent here for the 2 tasks, while also assuming that I have a linear covariation that links them between the 2. So that would, to give you some intuition about that, suppose in my task, my working memory regions, if they were lower or less active, I was also getting less activity in my auditory oddball, temporal lobe activity for whatever reason, okay? And so if that were the case, if there's a relationship there, this would be able to find it, okay? And there are lots of examples out there looking at different pairs of information like structure and function. So let's look at an example with EEG data using joint ICA. So what was done here was fMRI data was processed for an oddball task. So we got a contrast map out of SPM or FSL or AFNI. And then for EEG, we just took an event-related potential. So we basically took a time-locked average to all the target stimuli. These are done in separate sessions, okay? So we do a target event-related potential that looks like this here, okay? So the target stimuli occurred here, and then that's the average response following the target, okay? And now I have a pair of those for each subject, okay? So I have subjects by voxels and subjects by event-related potential time, okay? And if I analyze that with joint ICA to find maximally independent components, I get out something like this, where on the left you see the fMRI part of, so there's 6 joint components here. So these go together here. This is the fMRI part of this joint component, and the yellow curve is the same in all the plots. That's just the average ERP response of the raw data. But the blue is the component time course associated with it. So here's the one joint component, another joint component, etc. And you'll see it kind of separated on the EEG time course nicely into this sort of first peak, which is the N1 response, the second negative peak, which is the N2, sort of triphasic, and then these 2 parts of the P300. Yeah. Sorry, I can't hear. We need. Oh, it's not? But don't you know where I'm pointing without? Sorry, I have an arrow. I just have to change it to laser pointer. Yes. So here's the joint component. How long did you let me talk without? Okay. And so here's the first joint component, right? So you can see here's the N1, here's the N2, triphasic, first and second parts of the P3, and then a later response, okay? And then, so that sort of splits things up a little bit, right? In a way that, a region here that has a high value on the component. So this point in time in the ERP is correlated with these regions in the fMRI across subjects. Okay, that's how you interpret that. Okay? And then you can do cool stuff like for any point in time here, I have a weight for each of my ERP time courses, and I can go back to the fMRI maps and linearly combine them to get sort of a time, an estimate of what the fMRI signal looks like at that point in time. And so here, for example, if you watch, we'll step through this. Can I not? Yeah, so when the pointer is on, I can't, let me see if I hit the space bar. Whoop, that didn't work either. Whale, pointer options. Let's change it to arrow. Oh, anyway, I can't do it. For some reason, I can't undo my pointer here. But there's a real cool movie here, which if this white bar moves across the screen, you start to see fMRI regions pop in, and it goes actually very nicely between, you get initial temporal lobe activity when the stimulus comes on, then you get supplementary motor area, motor planning area, and then you get motor execution area, and then you see the P300 pop in. So you've got a spatially localized estimate of information without doing this inverse EEG modeling low resolution solution. And so it's a very nice example, and it's in that paper. You can see it in more detail. And you can also look at these at a subject-specific level, for example, to see where the joint components differ between patients and controls.
Sorry. In the previous slide that you showed, this is not combined EEG fMRI, is it?
This one is done in separate sessions.
Okay.
Here.
And so if it is not combined, I didn't get fully how do you correlate the time with the spatial maps? Could you expand?
So what I've got is basically a joint data set here, which has a map and a time course for each subject. Right. And then I'm estimating a joint component, and I have its covariation among subjects in the mixing matrix of that component. And so suppose I pick out 6 components here of interest, and for any point in time here, I have a component weight associated with each component, each joint component, and I can use those weights to linearly combine the fMRI map to get an estimate at that point in time.
Thank you.
Yeah. Okay.
Just to be clear, this wasn't trial by trial EEG. You did this on the average DR.
This was done on the average, although you could certainly do it trial by trial if you wanted to. But you'd have to feed in the trial data into the analysis up front, not do it after the fact. So, and that was our first study on that, right? And that was just using a central electrode in the EEG data. So we weren't leveraging the topology of the EEG at all. But you can do the same thing where you give it all the electrodes, right? And so that's what this paper was doing basically. So we get an ERP, we get an fMRI map, and we get EEG. A topographic map from the ERP data. Okay. Let's do another example. Again, joint ICA, but instead of giving it ERP data, I'm giving it auditory oddball contrast map and a Sternberg working memory contrast map. 2 different fMRI tasks. Calculate 2 contrast maps. I'm going to feed those in to my joint ICA. Okay. This one I like because if you look at this is the one component, when you look at you've got a loading parameter for each subject, for each component. So you can test for which components show group differences between patients and controls. And so this is the one component came out as showing a group difference, and it's this one here. But what I'm showing you here is once I have that component, I can go and I can calculate the joint distribution for oddball versus Sternberg, okay? So this here is a joint distribution for Sternberg versus oddball, and showing the controls in orange and the patients in blue. And if I just look down here at the marginals, which is sort of the integration down, I can see that the controls are shifted to the right, so they have more activity on the oddball task than the patients do. That's well known, published quite a lot. Other groups have published on that. But the Sternberg is different, right? It's got more variance in the patients, more strongly correlated and strongly anti-correlated regions versus the controls. And then the coolest thing is if you look at the individual subject joint distributions here, you'll see that the controls look much more flat, right? Each little cluster here is a different subject, okay? And the controls look much more flat. Their distributions kind of look like this, whereas the patients are kind of correlated between oddball and Sternberg. So what does that mean? That's telling us that the brain patterns associated with these very different tasks are more similar in the patients than they are in the control. So it's less sort of specialized to the task, which is kind of interesting, right? And it gets at a really interesting dimension that you really can't see if you look at these data sets separately. This is the joint information I was talking about. Okay, so sort of motivates why do data fusion, and just like we did with the group ICA approach, you've got all these hybrid methods you can do. For example, I can give it the information about patients and controls up front and tell it, find a component that maximizes that difference, right? And so we call that coefficient constrained ICA. And we can use that kind of an approach to sort of search through a bunch of combinations of tasks. So here, we just had, we have two different contrasts from an oddball task. We have a Sternberg task. We have gray matter data. And we have pairwise combinations or single maps from each of these. And we basically run a bunch of different analyses and sort of rank order the ones that are most discriminative of patients versus controls. And this lets us see, so this bar here, the taller the bar, the more it differentiated the group, okay, the patients and the controls. And so this one's showing that a combination of Sternberg and auditory oddball targets that pair is the most discriminative pair of information that we've looked at in this analysis. And you can use this to sort of say, well, okay, joint information, multimodal information is great, but there's ultimately going to be a trade-off between how much data I can collect, how long someone's going to stay in a scanner, and whether it's useful. So let's search through all this data and see what's most informative and what should we focus on, right, going forward. So that's an example of where we would do that sort of thing. And we did it again here, not looking just at contrast maps from a task, but also doing ICA and estimating, something like 15 or 20 components came out for each task, and we threw all those in as well. And we looked across all this stuff, right? And we can again see here that the ones at the top of the list here are the most discriminative going on down. And so it helps you sift. This is sort of saying, I want to sift through my data. I want to see what's most informative given a question of interest. In this case, we were interested in group differences. Okay? A complementary approach is canonical correlation analysis. So just to kind of explain what that is doing, what CCA does is it takes two matrices and it looks for sort of rotations of those matrices such that you could find maximal correlations, and it does it in an ordered way. So it finds the first most correlated relationships among those two data sets, then the next one, then the next one, then the next one on down, okay? And so it's a second order approach. It's not using independence, it's using correlation, but it again is sort of trying to say, I've got these two high dimensional data sets and I want to look at cross correlation, say, among subjects in those two data sets. And you can do this in a way that handles multiple sets. So in our case, a set would be a modality. So we can do fMRI and EEG and structural MRI, for example, and get out canonical representations of them. So in this case, fMRI map from oddball target activity, a gray matter map, and an ERP time course. And what we find, it's not always the case, but in a lot of the cases we find if we ask specific questions about sensitivity to group differences or ability to classify individual subjects, we do better when we incorporate some additional modality information into that analysis than if we worked with one modality by itself. Okay? And this is also true of, this is an MCCA analysis we did recently where we were predicting cognitive scores, right? So we had a whole battery of cognitive scores called the Measurement and Treatment Research to Improve Cognition in Schizophrenia. It's got 7 different cognitive domains. And we took all those and we tried to sort of predict performance on those different measures. And we actually did pretty well. For more than half of the subjects, we were with 95% accurate, and sorry, for more than half, we were 80% accurate, and in a subset, we're even more so than that. So, and then it gets really interesting because then you can say, given this prediction, which brain regions map into which cognitive domain, right? And this is getting to the So NIH is very interested in this research domain criterion, RDocs, which is basically saying we don't trust the patient labels and symptom-based categorization. Let's use some other ways to look at the data, and maybe it has to do with different dimensions of cognition, for example. Too late? No.
I just have a question. The combining multiple methods. Oh, you're going to talk about 3 methods, but in theory, right? You could combine ERP, structural, resting state, and task space, and get some sort of classifier out, right? That would maybe identify two groups, clinical versus healthy. And is there sort of an asymptotic trade-off by number of components we add into the joint, right? Like at some point, you could add in a lot of different data for a sample.
I don't think we're anywhere near that, knowing the answer to that question yet. You know, because we also have another huge problem, which is, as I mentioned early on, we're very, you know, dramatically undersampling the brain, right, with these imaging modalities. So you get some benefit, you know, in terms of the asymptotic, I guess this result here kind of gets at that, right? Here, right? When we sort of were rank ordering, if we throw multiple things in or less things in, which one's better? Pairwise, 3-way, 4-way, et cetera, right? So that sort of tries to answer that question. But I still think we need to throw more in at this point until we get better imaging modalities. So, yeah. There's also a trade-off with number of subjects as well. So if you have too many variables for any given subject, and say you had 10 subjects, right? That's not likely going to be able to be fit very well. So, yeah.
So related to that, you could also throw in behavioral or symptom measures and get components from that. But I'm trying to kind of wrap my head around the usefulness of that from the RDoC framework, for instance, where your clinical measures are fairly likely to predict caseness if you're, you know, looking at trying to classify schizophrenia from controls, for instance, because we've defined the clinical measures based on these clinical diagnostic criteria. So I'm wondering if you could talk a little bit about that and whether you use clinical measures in addition to these brain measures.
It's interesting. So it does a lot worse than you would think. Symptoms actually do a lot worse, because we use the SCID, for example, in the schizophrenia patients, this questionnaire to sort of diagnose them. But then if you look at like positive symptoms and negative symptoms in those categories, they don't predict very well. You know, symptoms are hard. They change over time, et cetera, hard to fully characterize. But we looked at, in this data set, we also looked at symptoms, and we actually were able to do a little better at predicting symptoms, even within group, which surprised us, actually. So we're still wrestling with that a little bit. But you know, I think ultimately a symptom is a downstream measure, sort of manifestation of a mental illness, which is probably not as specific as we need it to be. The hope is that, you know, cognitive scores and dimensions of things like that might actually help us do better. It may not, you know. So I think that's sort of an, you know, open question at this point. But, yeah. You know, and the other thing is, you know, you can use both kinds of information to look at. I didn't show any slides on this, but we're looking at sort of subgrouping, you know, within the data. So you have, for example, schizoaffective, which is kind of you know, a controversial category in some camps, and it's you know, schizoaffective can be, you know, either grouped in with schizophrenia, or, you know, you can have schizoaffectives that have, you know, one or more episodes of psychosis, and others that are primarily depressive episodes, and those maybe are different, maybe not, and so, you know, and that's symptom-based, right, in terms of defining it. So really, is the data that we're able to collect telling us or supporting that, or pushing against it? So. And in part, it does support it, and in part, it pushes against. So it's kind of an in-between answer, I think. Okay, MCCA and joint ICA. They're actually complementary to one another, because what MCCA is doing is really trying to sort of separate out strongly correlated things from less correlated things in the data sets. Joint ICA is assuming that we have a mixing matrix that's in common across the two modalities, right? There's a linear relationship between the regions or the time courses in each joint component, okay? But it's using higher order information to identify those. So you can combine the two of them. You know, if we didn't have enough letters in our acronyms already, now we can do, you know, MCCA plus joint ICA, okay? And so there, what we do is we use MCCA to find these latent variables in our data and essentially help us order them by correlation, and then we do a joint ICA on that linked, you know, data set to maximize independence among them. And so, you know, I'll leave the details to the papers here, but we did, you know, we've done a bunch of simulations, and we show in a lot of cases MCCA plus joint ICA actually helps mitigate the disadvantages of both approaches, and actually does well in both cases. And using this kind of an approach, you know, we, for example, did a study that involved, I think this was using ALF maps from fMRI, and then this was using fractional anisotropy from a diffusion weighted imaging experiment, and then gray matter maps from structural data. And then what we see here, for example, is here, so here's the fMRI, here's the diffusion, here's the structural, and then you'll see that there's either 3 or 2 or 1 row in each column, right, that's got an image in it. And that tells you whether we've got a 3-way relationship among modality. In this case, all 3 of these sets of regions are linked together in the model. And this one actually shows, you know, and you know, you can look at sort of how they're related across the different mixing matrices. And then in this case, we had only diffusion, right? So there wasn't evidence in the data to support that component being linked to the other modalities. So it's able to find, you know, sort of modality common and modality unique information, which is really, I think, important, because, you know, we don't know, right? This is really complex, right? And it's hard to know. It's even hard to formulate what question would you ask, right, up front. I think hopefully that will become more clear as we continue to look through these data and inform us about the questions we should be asking going forward, right? But this really lets us look in a way that doesn't assume there's a link between everything. There might be or there might not be. Okay? And another plug for the benefit of simulations. When you're talking about simulating, you know, it's always challenging, because you can simulate things in so many different ways. But when you're looking at multimodal stuff, you have to start to simulate sort of joint information across modalities, right? And so that can be tricky, right? Most of the studies out there just sort of assume a little linear thing and then throw in and do some ad hoc comparisons. We've played around with sort of a statistical framework to do this, where we're using what's called a copula, which is the Gaussian copula is what caused the stock market crash a few years ago. One of the things, because they were looking at joint information among these hedge funds and other things like that. I don't want to cause a crash in our brain imaging data here, but we can use that as a model to basically generate data sets that are statistically valid in terms of the joint information, characterize it better, and then see where these models break. Okay, so just a plug for that. I think there's still a lot of room for better simulations. So in the last portion of this talk, I want to talk about imaging and genetics. Have you had any genetics so far? Okay. So, you know, but you can think of it as just yet another piece of information. But you can actually get information out of these chips with a saliva swab or with blood. You can basically get sort of a sampling throughout your DNA of what are called single nucleotide polymorphisms. So basically a single base pair here will have a mutation that varies across subjects. and it can be either homozygous or heterozygous. And that information in some cases is linked to disease. And some of it is normal variation, right? And so you basically, for each of the samples, you get out a number, 0, 1, or 2, whether it's homozygous in one or heterozygous in one or the other direction. We can also look at larger chunks of DNA called copy number variations that are either inserted or deleted, and things like epigenetics, which basically are control the degree to which your DNA and your genes actually produce downstream proteins or not. And so you can sort of have that turned on or off by these methyl groups attached to your DNA. And the cool thing about epigenetics is it can change over time. So your DNA is fixed unless you're in the X-Men movie and you get hit with some cosmic ray and your DNA changes, right? Pretty much fixed for your whole life, whereas epigenetics is changing, right? Smokers have more, on the whole, a lot more methylation, for example. So this data is a little bit, in its characteristics, is a little bit different, right? With the imaging data, we've got sort of continuously valued information. This is a little bit different. So we treated this sort of with a little bit of a looser assumption, using an approach called parallel ICA. So in that case, you sort of imagine on one side we've got fMRI, which again is this feature-based approach where we have a contrast map, for example, subjects by contrast map. On the genetic side, we've got subjects by the code, you know, the 0, 1, or 2 data, right? And what we're doing is really trying to do two things. We're doing ICA on each modality, fMRI or SNPs, and then jointly taking the loading parameters from those and putting a constraint on that to sort of maximize the correlation, but not for all of the components, just for a subset of them. Okay? So again, it's kind of a hybrid of the things we've been talking about already, but you're basically you're trying to maximize independence and look for links between these data sets. And you know, using this kind of approach, we show that it's actually more sensitive than massive univariate correlation, which is what's done quite a lot in imaging genetics, where you sort of have your favorite brain region, and you have a million different SNP markers, and you just do a million different correlations, right? Or you have a SNP, and you look at cross-correlation among all your different voxels, right? So again, that's the voxel-wise approach I mentioned at the very beginning of the talk. So just to kind of show you what this looks like, if we take a data set, again, of patients and controls, and they have a task, an fMRI task, and we have a SNP array that comes out, you know, we got out of that data set basically a component that showed a strong link between fMRI and genetics. Here is a picture of the component. This was actually a sensory motor task, so it looks like the task-related component is actually the one that was showing a link to the SNP data. And then for the SNP, so if you thought interpreting brain regions was difficult, genetics is much worse in some ways, probably because we know less about the brain than we do about genetics thus far, because they're ahead of us. But for every SNP, it falls within one or more genes, because there can be some overlap, and then each gene has downstream implications in terms of biological pathways and proteins and things like that. So you have all these databases out there where you can give it a list of sort of the top-ranking SNPs, and it can say these are the biological pathways that are, you know, you can infer from that, and that can help you in sort of interpreting whether this makes sense. So, for example, in this case, looking at schizophrenia, you know, we had actually at the top a lot of SNPs that had been previously implicated in schizophrenia, which was actually nice. But then we had some others that were related to neurotransmitter signaling that had not been previously implicated. Again, giving us some targets that we can look at in the future. And you can have scenarios in the parallel ICA algorithm where you have, you know, certain components. You might have two fMRI components linked to one SNP pattern. A weighted combination of SNPs might relate to two different fMRI components. You can have a one-to-many kind of relationship there, which is another thing that's nice about this approach. In this case, we had one that was actually showing a group difference, and the other one was not showing a group difference, suggesting it was sort of similar in the patients and the controls both. And just as in joint ICA, where we looked at ERP and fMRI, we can look at ERPs and genetics, right? And so in this case, we have an auditory oddball task, we have an event-related potential for each subject, and then we have an array of SNP information for that subject. And we fed the target ERP and the SNP array into a parallel ICA analysis. And we also did the same for the novel stimuli. So for an oddball task, you're basically getting this sort of infrequent high tone. And that elicits what's called an oddball response in your brain. You're supposed to press a button. It's a salience task. And then you get novels are these other infrequent stimuli that are sort of like doorbells and train whistles and things like that that are non-salient. So you're not pressing a button, but they both elicit an oddball response. And so we took ERPs from those two different stimuli, and we looked at them with parallel ICA, and we got out, here's the fMRI, or sorry, the ERP side, and then here is the genetic side. And what we found was that the associated SNPs were almost the same for those two very different, stimuli, which was actually, we thought, a very nice result, because they were both, the SNPs, if you fed these SNPs into a biological pathway database, you would get, you can get something like this. And what we found was adrenergic and dopaminergic pathways were primarily implicated for both targets and for novels, which are both involved in this oddball response. So that was kind of a nice result. You can do additional things. So one of the problems, obviously, is we don't have enough subjects, right? Because we get more and more variables per subject. We have, we can do millions of genetic markers these days. But typically you're dealing with hundreds, or if you're lucky, thousands of subjects. And very few studies have gotten beyond that in terms of the number of subjects. And so you can actually feed parallel ICA, again, a constraint. So you can do a sort of a hybrid analysis where you give it, maybe you're interested in a particular pathway. So you go to the database, the informatics tools, and you generate a list of genes and SNPs that are implicated in a particular pathway you're interested in, and you can feed that reference to parallel ICA. And one of the reasons this is nice is because you're not sort of assuming that there's no other SNPs involved in this pathway or no other genes involved in the pathway. You're letting the data tell you that, but you're adding this constraint which helps sort of regularize the data. What it does is it, you get more power per subject in your data set because you've constrained it a little bit more. Okay, that's kind of the trade-off that you have there. And so this is one analysis we did where we used this so-called ONC pathway as a sort of reference set going into the genetic pattern, and pulled out these associated regions from a structural gray matter data set. We're almost done. Almost done. It's getting warm in here again, I think. In addition to that, you don't have to stop at 2, right? You can do 3 ways. So, in this case, we have fMRI, structural MRI, and SNP information. And, we're very interested in things that basically tell us, how proximal to the actual behavior is the information that we've measured. So obviously, your DNA doesn't change, right? But it kind of is your ultimate prior, right? And then you have epigenetics, and then you have gray matter structure, brain structure, and then you have brain function, and then you have behavior. And so this kind of a sort of hierarchical analysis, we can look at this kind of thing with sort of multi-way parallel ICA, and we can get out basically maps from that indicate certain or implicate certain regions that are showing differences in brain volume, regions showing differences in activity, brain activity, and also certain SNPs. that are tied to pathways. Okay? All right, so now we're in the last section here. And I know this is a lot, but you have the slides, and so, hopefully this, I'm kind of throwing a lot at you so you can kind of see there's been a lot of stuff done, and hopefully it's not too opaque as we go along. There's fewer questions being asked, so I'm not sure if you guys are getting ready for sleeping out there, going to sleep, or eating lunch, or what. What's that? You're getting your pictures taken. Oh, really? Where? Okay. And so we can combine the concept of this joint analysis, as I mentioned, where you have sort of features, with the group ICA approach, which is sort of looking at within multiple subjects, you might have time by space, have a matrix for each subject, right? And so you can bring those things together where you're trying to do a parallel group ICA, right? And so here's one example where we had concurrent EEG and fMRI data, so collected at the same time. Now we have two different matrices and multiple subjects, and there's a relationship in time, between the two that we can sort of exploit. So we basically have two different group ICAs, one on the EEG and one on the fMRI data. We're going to talk about the GIFT toolbox in a minute here, but there's embedded within that, there's another tool called EEGIFT, or eGIFT, which is an EEG group ICA toolbox as well. And this lets us pull out, for example, fMRI patterns that are associated with certain, certain changes in EEG frequency spectra. Like, for example, here we've got the different bands going from delta, theta, low alpha, high alpha, etc. And then we're showing the brain regions that sort of came out or were sort of linked to those patterns. I'll leave the details of this for that paper that I've cited there. And then we can also ask questions, for example, about intrinsic networks and brain structure. So in this case here, we have a group ICA done on fMRI data, and so each the blue one is the fMRI data. And so it's paired with its associated structural gray matter covarying component, okay? So you'll notice that basal ganglia networks in fMRI are, matched pretty well with gray matter changes in basal ganglia. So left and right basal ganglia covary together in structural data and in functional data, okay? And we typically see things where we get one component for structure, like here we're getting the visual cortex. It's fairly broad, right? And then we get three functional components that sort of match closely to that. And so what's this telling us about the gray matter? It's telling us these gray matter regions sort of covary together. The visual cortex covaries together. It tends to go down or up together across subjects in gray matter, okay? And you can, just like with fMRI, you can build up networks and look at a cross network or a cross component links in structure, just as we did with function. Okay, and then just the last two slides kind of go into some other different domains. So in this case, again, we have a group ICA model, but what we've done is, instead of giving it just a feature like a fractional anisotropy map, what we've done is we've calculated the all-to-all probabilistic connectivity matrix, right? So we haven't reduced it down to this summary measure. We've got this sort of very large, sparse matrix for each subject, and we can feed that into our group ICA model and essentially do a data-driven parcellation of the diffusion data using group ICA. And so I'm just showing you a small example of the corpus callosum, and notice how nicely it sort of parcellates here, the corpus callosum, using that data. It's a completely data-driven parcellation. We haven't used an atlas to do this. We haven't used a seed to sort of look at A to B. We've done all voxels to all voxels. And we have information here in the gray matter as well. So we can look at very, very low or weak diffusion changes in gray matter across groups or across components, etc. And then the last slide is just sort of demonstrating, we and others have started to do this also with MEG data, right? So you can do, in MEG, you have, again, a bunch of sort of squids, magnetic sensors around the head, but you can do this sort of more sort of focal reconstruction approach called a beamforming approach, and then take, and you can do different kinds of filtering on it, and you can then do essentially a spatial ICA on the MEG data to get out, again, sort of these networks here that match reasonably well. Again, you notice that the MEG ones, they're bigger, they're smoother, a little blurrier, but you can get pretty good matches. And this is building on work that Matt Brooks did early on. And so again, you know, where are we headed with all this? I mean, if you ever do take sort of a hard look at, I've got fMRI data and MEG data, I'm going to just, let's have one person do a group analysis on this and one person do a group analysis on this, and let's compare what the answer is, right? You get, you know, uncomfortably different answers when you look at these two data sets, right? The fMRI on the whole shows decreased connectivity in most places, not all, for schizophrenia. Whereas in MEG, you get a lot of these strong anti-correlations, you get a lot more frontal activity that are showing changes in the schizophrenia data sets. Both of them are finding kind of similar regions, but you know, ultimately the truth is somewhere integrated across, you know, those data sets and whatever bits of information we haven't been able to collect from these living people. So I did, and this last one, okay, this is the last one, I promise. Okay. The last one is just a sort of a teaser slide to say, you can actually, all these are linear models. We've been doing linear models all along. We're actually looking at some nonlinear stuff using something called deep learning. And you can get at things like nonlinear ICA, and you can get at, you know, sort of more interesting relationships there. And so, for example, there are these models that will do, they're called recurrent neural networks, and you could sort of do, you know, Google does this and Amazon does this. They're trying to do translation, right, between English and French, by training these models on these large databases and then getting you know, a model that sort of looks at the context and the relationship among words. And so we're using these kinds of approaches to try to look at the relationship, for example, between, you know, these different modalities. For example, even brain structure to brain function. It looks promising. I think that we're getting at a point where we have enough data and we can actually simulate realistic data that can help us train these models. This is a lot more parameters than what we had with even ICA to get out answers that are actually reasonable. So that's it. I am done now. We have all this software here. We're going to talk about some of the tools within the GIFT toolbox. I'm not going to go over the fusion toolbox, but all the approaches I mentioned, the parallel ICA, joint ICA, MCCA plus joint ICA, all that stuff is in this fusion toolbox, and there's a manual for it and a walkthrough, so you can go online here and play with it if you want. The simulation toolbox, and I mentioned this neuroinformatics software. So this is a picture of of my lab, and they're the ones that are making all this work happen. And I think at that point, are we going to take a break now?
We're going to
and then
maybe
jump
a
into
question or two and then take a break to take photos?
Okay, sounds good.
So just a real quick question. So it seems to me something would be interesting in a sort of a longitudinal treatment-based study is to look at something like change in multiple methods to predict as a classifier who ends up getting placebo versus, you know, the active agent, or who ends up improving in treatment or not. Can you talk at all to how complicated or easy that would be in this framework?
It can be done, and we've looked at like predicting in some initial cases, sort of treatment response, or even patient groups that sort of decline. Like we've been looking at certain disorders that basically have folks that either rapidly or slowly decline, and so yes, you can do those sorts of things.
I have a question about joint ICA using EEG and fMRI.
Yeah.
So from my understanding from the way you were talking about the components, it sounds like you get a component which, say, relates to the N170, and then you can look at the fMRI component to see where the N170 would be located. And if that's correct, then I was wondering how to sort of conceptualize that when the fMRI data is only being collected every 2 seconds.
Mhm.
We don't actually have the fMRI data for the N170, or can you
Yeah,
talk
so
about
I would state it differently. So what Joint ICA has done is it's found fMRI regions that are linearly related to certain points in time in the ERP data, right? So it's used the covariation among subjects. So there are certain regions in the fMRI that correlate with certain time points in the EEG. And so it's just kind of, that's what it's trying to do. Interpreting it is a whole nother thing, right? And so what I was showing you with some of the work that we've done is that we get these sort of sensible results, right? So for example, it made a lot of sense that for an oddball task we saw in the reconstructed fMRI maps, we got auditory first, then motor planning, then motor execution, then oddball response, right? And I think that you know, the fact that you've got, it's basically leveraging covariation among subjects and enabling us to sort of make, it's a different kind of inverse modeling, so to speak, but it's sort of forward modeling instead of inverse modeling.
Thank you for that talk. You know, I've seen you a couple of times, and it fascinates me that every time I see there's something new coming out.
I'm surprised by that. No, I give the same talk every time. So.
Yeah, it's so fascinating. It just fascinates how far the ICA can go. But I'm going to ask you something at the very basic level. ICA is sort of based on, you know, that finding the independent components is by maximizing independence through you know, finding basically non-Gaussian signals. So it seems like it works really well for most of the biological signals, including fMRI, which are mostly super Gaussian Laplace-like signals. But have you seen, being a devil's advocate, what are the problems with ICA? When can you actually not?
There are no problems. Shh.
In what situations would you say ICA fails? In the sense, if you have a really nice, like a Gaussian signal, it seems like you wouldn't do such a good job at really getting these components out. Right?
Yeah, so the ICA algorithms that have been applied to fMRI data have largely been fairly simple, sort of linear ICA, and also sort of assuming unimodal distributions and super Gaussian distributions, right? And so those like fast ICA and Infomax, those algorithms by default, I didn't really talk about this too much, but you've sort of picked up on it. By default, they're trying to maximize independence, and you can't uniquely separate two Gaussians from one another, right? So in contrast to the GLM, which assumes that you have Gaussian noise, in ICA you're typically assuming we have non-Gaussian signal, which is, you know, usually true. But if you want to separate two Gaussians, if that's your question, then you need to incorporate some variations on the algorithms, and there are algorithms that can do that. Other places, so again, you know, I would never call ICA a model-free approach, right? Because it has an embedded model, and anytime you deviate from those model assumptions, you would, you know, you can potentially break it, right? And so if you have things that are nonlinear, right? That's why we're starting to look at some of these nonlinear ICA approaches to see, you know, there's a lot, you can model a lot of things with linear models, right? Especially combinations of linear models. But it looks like there's some cases where there are some nonlinearities that are picked up better if you incorporate nonlinear models. The other thing is all these, you know, it's ultimately sort of an analytic specter, right? You've got, I'm just going to assume in the extreme case that I know what the relationship is, and I'm going to test for that relationship, versus the data is going to tell me what's there, and then I'm going to look post hoc at what came out. And then I've tried to sort of, you know, reflect this in my talk, right? And then in between is constrained approaches that are sort of assuming a little bit and giving the algorithm a little bit of that information. Where we should be on that spectrum, I don't know. I think we're getting a lot of useful information. I think some of the ways you can break it would be, again, as we showed in the simulations, if you start to deviate extremely from similarity across subjects, you can still you start to see some attenuation, effects there, and there's probably others, but yeah.
Thank you.
Yes, and we're going to do the GIF demo, so I can answer a few questions kind of as we're doing that. So.
Get your slides up.
We got the PDFs just now.
And I'm pretty sure I have your slides online already, right?
They just got it from me
Oh,
like
good.
a minute ago
Oh,
or
good.
so.
Oh, so you're still working till the last minute?
No, I wasn't working on that. I was just like, for some reason, I can't connect to the web. For some reason, my mail are not going out of my outbox, so I didn't send it. I'm on
which
the
ISP
UCLA
is it?
web.
Yeah, it won't.
That one is.
That one's
That's
blocked.
interesting.
It's
That's interesting.
what you
Why
have
is
to
that?
use is the EduRoam. Excuse me.
Okay. Well.
Circle around it.
Oh, okay. Well, that's. I mean, it's fine. I don't think anybody would have looked at them any earlier anyway. So. Oh, directly. Okay. I didn't. Yeah. Wow. Exactly. It's like.
We had a breakthrough here.
Okay. All right. Introduce the topic? Yeah, I can just start talking and get people to their seats. Thanks. So we'll be talking a little bit more application-oriented today as compared to yesterday when we focused on the different methods in graph theory. So I took the freedom to turn this talk a little bit into a hopefully more, if you will, inspirational talk in the sense of actually posing or creating more questions than it answers. And in terms of understanding how important connectivity analyses have become, how important this view on network interaction has become in neuroimaging and in neuroscience in general, and what I think the probably most exciting still open questions are. So at the beginning, functional imaging was very much focused on localizing specialized functions in focal areas. And we did a whole lot of that for a very long time. And then we realized that a lot of cognitive functions actually emerge through the simultaneous activation of a whole set of remote regions and large-scale neurocognitive networks rather than single areas. And then there is still basically discussions going back and forth about some of these higher order networks in terms of what their specialized function is. But of course, this is also still a localization point of view on the brain. Instead of asking about functions of single areas, we're now asking about functions of large-scale networks. But really the big paradigm shift in the last 15 years or so has been the understanding that cognition arises from the interaction, often measured as connectivity, between brain regions, and also at smaller and faster scales, of course, in smaller networks of neural populations. So this is already a few years old, but you see the PubMed search for functional imaging and activation in this beige color, and functional imaging and connectivity. And I think that this curve must have continued more than exponential in the last 5 years. So I'm just going through that same idea in a little bit more detail to try and illustrate where I think the field is at the moment. So this is one typical example of the early approach where we're asking, okay, how can we maximally well localize the specialized function that humans have or primates have in general, face recognition and recognition of individual faces and such. So what the clever fMRI designs usually do is having some sort of a task that requires the cognitive condition of interest, the function of interest, which is in this case face processing. And then we're trying to find a maximally well-matched other condition that matches in terms of all cognitive demands except for this particular specific function that we're looking for. So in this case, you would contrast processing of faces with processing of other very specialized and very often observed objects. So it's this special category of objects as opposed to other objects. And when you do that contrast, of course, you come up, you detect the fusiform face area. But we all know, all of us who have done functional imaging, that if you were to look at the main effect of the task across a more passive baseline, you would see a massive amount of activation all across the brain, both in very functionalized areas as well as in these higher order large-scale areas. And that's not only true for this kind of complex processing, but it's also true for very, very simple tasks such as detecting a simple sinusoidal tone. If you look at the main effect in the fMRI image of what is happening in terms of network communication in your brain just to detect a simple tone, it's absolutely astonishing. So one of the really, I think, early occurrences of this idea that functions don't occur in specialized regions necessarily, but maybe, especially for higher order functions, a result of activation, co-activation of a whole set of regions, came from the Corbetta lab, where they characterized the dorsal and ventral attention systems. In this case, you see a typical experiment that would result in activation of the dorsal attention system, where the subject is requested to covertly attend to the right or left area in the space, and you see intraparietal activation, frontal eye fields, middle temporal areas. All of those together are necessary to bring about this cognitive function. And then in the last 10 years or so, there was really this shift in realizing that these same areas, in this case the visuospatial or dorsal attention system, these areas not only show co-activation when the respective cognitive challenges arise, but they co-activate all the time in synchrony, even when we're resting, even when we're unconscious. And that's really the amazing part. It has not even that much to do with conscious mentation. It is really this continuous correlation of high-level activity. And I will close the talk at the end by really pointing out that it is the big open question of trying to find out why we're spending so much energy in the brain on trying to keep this kind of activity up, even in the absence of evident cognitive requirements. So from this realization, of course, we moved forward very quickly to try and understand how the regions within each one of these networks correlates with each other as a measure of communication, and then also trying to understand how these networks communicate basically across the networks, right, across the entire system. And we talked a little bit about this kind of graph approach yesterday. I'm going to more focus now today to the move from this static view of graphs over the entire session to the dynamic functional connectivity, where here you see a functional correlation matrix. But if you were to break down the same session into discrete time windows and repeat your correlation analysis there, you would actually see that a lot of these windows look nothing like the average window. In fact, functional connectivity is very dynamic over time, and we heard that this morning as well. And I'm going to focus on,
Given this intrinsic brain connectivity, regardless of the task, in those studies that we do, like network analysis, when some task is present, when we are showing like some stimuli, do studies take into account the intrinsic connectivity and take that out? Because this is not a stimulus driven or task driven.
Right.
And that may confound like all the network analysis. So I'm thinking how one might take this into account or regress it out some way.
Good question.
Or do people do that as well?
So, of course, just because we didn't model this intrinsic connectivity, it doesn't mean it wasn't present here, right, for the first 2 decades of fMRI, right? We just, most of neuroscience treats intrinsic activity as uncorrelated or white noise. We're assuming that noise term in the end of the GLM somehow magically takes into account this kind of noise. But of course, this is, as we know, very remote from what the brain is actually doing, because this noise is not white at all. It's very, very structured in time and in space. And when we do this kind of an experiment, we repeat our trials a lot of times, and that's how we can model them over and above the background fluctuations, which are in most cases much stronger than the evoked activity. So if you're looking at raw data, take a look at your raw fMRI data. Just take the time course out and mark where the events happen. You're going to be really, really shocked if you haven't done that already, and how much stronger that background fluctuation is. Even better if you have a method better than fMRI, like an electrophysiological method where you are a bit closer to the neural activity. So to come back to your question, there was one study out of WashU where they actually showed a very strong similarity of seed-based connectivity networks when they are derived in the same GLM as where you're putting in your evoked regressors. So if you were take any random fMRI experiment and put in your task evoked regressors, if you add any time course of a particular node as another additional regressor, that time course is going to pull out, in addition to your task regressors, that particular connectivity network, because it's still present during task, right? And you can do that with any network, not only those that are important for your task, obviously. So if you should take it out, I actually started adding compartment regressors to my standard fMRI experiments. So in the GLMs, I actually do have regressors that take out, for example, global white matter or some derivative thereof, like you guys heard about ComCore, if you want to be a little more conservative and cautious, or even global gray signal. That's a little tricky, though, global gray signal and task evoked activity, because yeah, I don't want to go into too much detail right now about that, but I do think it's a good idea. It cleans up the data if you do take out intrinsic activity in some way, but be conservative and not taking too much gray matter activity out of it. I have seen my contrast improve radically by doing that.
Thank you.
Sure. And so lastly, I want to move on from the realization that function connectivity is very dynamic to the interaction between each one of these dynamic states and this task evoked activity, because this is where I think really cognition is emerging. It is in the emergence of the intrinsic state of the brain with task evoked activity that cognition is happening. So this is really what we need to study a lot more than we have done so far. So I'm hoping to really close this loop a little bit today, at least. I'm probably going to open up a lot more questions. So you guys are an impressively international crowd. How many Germans do we have here? None. Any Swiss people? Phew. Okay. So yesterday I introduced the father of graph theory, Euler, as a German mathematician, and I quickly realized he's actually Swiss. So where I'm going with this is I have a bias in this talk, right? So of course I'm organizing it by my own passions a little bit, so please excuse me that the last several slides are our own work in large parts. That's of course because I'm passionate about the questions that I'm studying, obviously, but also because I went through all of those questions that I was pointing to yesterday in terms of the decisions I had to make along the path of applying graph theory. And since I went through those myself, I might be able to illustrate a little bit more why I made certain choices. So I hope that's going to be helpful. I'm going to talk about dynamic functional connectivity and how functional connectivity moves beyond structure to some degree, and how that is related to the understanding that functional connectivity is actually non-stationary. And then I'm just going to give one example that you actually saw earlier today, in the sense that dynamic connectivity may allow you to distinguish between healthy and non-healthy brains in a way that static functional connectivity would not be able to. I will then go to this probably slightly more philosophical question of how functional connectivity is actually different between rest and task, and whether it's actually a good question to ask that. Maybe it's not the right kind of question. We will see. And then how intrinsic connectivity and activity actually interact, and how that may actually influence behavior. All right, so you may have seen this last week. In this study, the major question was, and how far functional connectivity really reflects the structural connectivity of the brain. And so you're seeing about 1,000 regions of interest averaged. The data is averaged across the same 5 participants for structural and functional connectivity. The structure was derived from diffusion-weighted imaging, and the regions of interest are ordered from anterior to posterior, that you can appreciate a little bit the clusters that the functional connectivity networks may build. So this comparison was quantified down here, where you can see the strength of the functional connections as a function of the structural connections. And over these 1,000 regions of interest, there was a very strong correlation between the two. And they repeated the same analysis, concentrating only on the default mode network. So here all the red dots are regions that in the structural connectome were connected to the few regions of interest that, or the seeds that they chose for functional connectivity in the default mode network. And then only a subset of those 1,000 nodes, the 200 nodes that belong to the default mode network, show this effect very, very clearly as well. So here on the left is the structural connectivity from the default mode network that I showed you in a different view just now. This is the resting state functional connectivity in the same participants within, again, the same nodes of the default mode network. And then what they did was simulating functional connectivity on the backbone of structural connectivity without looking at the actual functional data, and then try to predict what functional connectivity should look like if it was directly derived from structural connectivity. And you see quite a bit of spatial similarity. And here they're plotting the the modeled functional connectivity as a function of the observed empirical functional connectivity for the regions of interest of the default mode network, and they see a high correlation. But there is some surprising deviations. For example, the lateral inferior parietal areas of the default mode network are not actually coming through in the model very well. So this is where it gets a little bit more interesting. They are plotting here the strength of functional connectivity, or how often that was observed for region pairs where structural connection was actually present versus when it was absent. And you see that the two functions are not very well dissociable. The two distributions are not very well dissociable from each other here. So in the simulated data, the two distributions are actually much better dissociable. If you plug in the same data into a different viewpoint and look at the receiver-operator curves, this difference between the actual empirical data and the simulated data becomes even more clear. So what you're seeing here is across different threshold of functional connectivity, if a functional connection is present between a certain set of brain regions, how probable is it that that connection was actually really present in the structural data? So this would be true positives, predicted structural connections that were really present, and false positives, predicted structural connections that didn't actually exist. And the prediction is not doing very well in the empirical data at all. So the region under the curve is a lot smaller than for the simulated data. So they conclude that structural connectivity strongly shapes functional connectivity, but you cannot really infer structural connectivity from functional connectivity all that well. So they're not really equivalent to each other. So some more simulated studies in this case, I think Randy McIntosh beautifully discussed this with you guys. So if I'm giving you too much repetition, please just let me know. So in this case, Gustavo Deco and Randy McIntosh's group and other groups have simulated functional connectivity on the backbone of ground truth, in this case for macaque monkeys, or a realistic structural backbone where we know the ground truth. So you can, for example, look at the COCOMAC database, which is a very beautiful meta-analysis that gives you information across all the tracing studies of macaque monkeys. So based on that structural data, they report that it is enough to model noise, some level of noise activity in the nodes and conduction delays in order to observe the spontaneous occurrence of fluctuations in activity, and that includes infraslow fluctuations in the range that we observe it in the bold signal, but also transitions and functional connectivity across time. So this falls out of these models without even putting in anything extra other than these two requirements, which is interesting. So I'm just citing directly the sentence because I think it captures really an important point, that resting state networks, network dynamics is the gateway to the array of cognitive architectures that the brain has available. In a sense, resting state networks represent exploration by the brain of what is possible in terms of cognitive setups. So this is all simulation. Now let's look at what the actual empirical data is doing in this field of dynamic functional connectivity. So this is a slide from earlier today. So if you have any questions, Vince is here. Yeah. So the idea that functional connectivity changes over time has most often in the literature of the last few years been studied using sliding window approaches. And there are a few other approaches. I think we saw time frequency analysis earlier today. So there are other ways to go about this, but by far the most common analysis has been the following. So I'm actually directly showing the pipeline from this particular paper that you saw earlier today. They have actually a very nice high number of participants, a little over 400, and these are 50 ICA components selected out of 100 that they started out with. And then for each subject, they're extracting the time course of these nodes. And instead of calculating the correlation matrix across the entire run, they are actually using a window of 44 seconds. That's in their case 22 volumes. And they're just calculating the correlation matrix here and slide that over time. So you will have a sliding window snapshot of connectivity, but calculated over much fewer volumes, but that gives you some sort of a temporal resolution. And then they aggregate this over all the 400-something subjects, and they apply a clustering algorithm, in this case with a K of 7, to automatically subdivide all of these connectivity snapshots across the participants into more or less discrete states, if you will. So this is a little bit along the ideas of what Randy McIntosh was calling multi-stability, as opposed to having a continuously fluid changing connectivity that never reoccurs. This analysis is trying to take the idea that some of these states in more or less the same shape, at least to some degree the same shape, are iteratively revisited by the brain. So here are the centroids of the 7 different cluster states, basically. And on top here, you see the percentage of time windows over the total number of time windows that each one of these states was observed. And so that varies vastly across these states. And then another interesting thing is, given that they had so many participants, they could actually look at the evolution over time on how often this particular state was observed at a particular volume and time. And so it's interesting to see that this state, for example, becomes more and more and more frequent, while this state actually becomes less frequent over time. Another point that they observed is they actually applied modularity on these matrices. And you see here the modularity metrics denoted by Q. And you also see the number of modules here by different colors. So the number of modules are different across, these states. And the strength of modularity also varies. So they observed a fluctuating amount of modularity over time. All right, so this is another study that was looking at windows at 43 time points. These are a lot longer. Sorry, they're longer in terms of the number of volumes, but what's special about the study is that they had higher temporal resolution. So they're below a second. They're at 720 milliseconds. I'm not sure how much they actually gained from it, given that they took still quite long time windows. There's no direct comparison with slower imaging. I think the point that was made earlier is, I think, a valid point that if you can image faster, it's mostly going to benefit you in terms of being able to recognize the aliased basic physiological noise components, rather than really gaining anything in terms of neuronal function, because given the HRF itself is too slow for that. Anyhow, so they have this faster resolution, and this is for two single subjects from the Human Connectome project, actually. And so what they observed is they're measuring, let me get that actually. They're measuring a measure that they call nodal efficiency, which is basically derived from the mean inverse shortest path length of a node. So this is similar to the global efficiency that we saw the other day, except that you're looking for every single node separately in this case. And the reason they did that is that they are plotting here nodes and time, and the heat map is basically the value of that efficiency. And what they're pointing out is that efficiency seems to change at certain time points across a very large proportion of the nodes at the same time. So there is this kind of group movement into a different state and back. And another thing that they point out is that the onset of the high efficiency nodes, at least in certain participants, is very sudden, while it tapers off more slowly. So these are basically maps of efficiency for a very high efficient and low efficient time point, just to compare whether there is a spatial distribution to it. And another observation that they made was actually also reported by several other studies already, which is that if you apply modularity, in this case they got 4 modules out of their data, and then look at which nodes actually change functional connectivity more than other nodes, then you find that the nodes across modules are the most, the least stable, the most dynamic, and the nodes within the modules, at least for most modules, are the most static, which is no surprise, because otherwise if you did the whole session, then everything would be connected with everything if the across module connectivity would not be dynamic. I guess it could just be completely absent. It is an interesting observation that it is not completely absent, it's just going all over the place. So for example, one area that was this was observed was this big discussion about anticorrelations and whether they are real or not. And it was observed even in electrophysiological data that there is true anticorrelation between so-called task positive and task negative areas, but that these networks go in and out of sync over time. So there's a percentage of time that they're actually anticorrelated, while at other times they may even be positively correlated to each other. So connections between modules are highly non-stationary. And then another study that I believe you also may have seen earlier today is how we can apply dynamic analyses of networks in order to dissociate maybe differences between healthy controls and people with disorders in a way that static analysis may not be able to show us. So here you're seeing actually the correlation matrix for healthy controls and schizophrenic patients. This is actually not the dynamic analysis yet, so this was calculated across the entire session. And when you look at the direct contrast of the two, you see immediately a strong hypoconnectivity within the sensory areas. So not only within auditory, visual, and somatomotor, but also across those networks, there is an extremely visible drop in connectivity in the schizophrenics compared to the healthy controls. But additionally, you see hyperconnectivity between certain subcortical areas. So I don't know if you can see this white box here, but it's denoting the thalamus that is hyperconnected to these sensory areas in the schizophrenic patients compared to healthy controls. So here's the dynamic analysis. So in the top row, you have the healthy controls, and here in this row, you have the schizophrenic patients. This is the same analysis that I showed you in the other paper. In this case, they chose 5 clusters, and here you see the direct contrast between the two populations. And what they're pointing out is that the hyperconnectivity between the thalamus and the sensory areas is actually occurring only in these 2 or 3 states. It's not constantly the case. Additionally, they saw what you see here in the colder areas, a reduced connectivity between another subcortical area, the putamen, and the sensory areas during the same states. And here you can see the so-called dwell time. This is the amount of time that the subjects on average spend in any of these kind of states. And you see that the healthy controls and the schizophrenic patients are very different from each other, and that the schizophrenic patients don't actually spend as much time in these states 2 and 3. So what they point out is while they could observe this point about the thalamus hyperactivity already in the static analysis, the hypoactivity with the putamen was not visible without doing this sliding window kind of analysis. And this has been applied to other disorders as well. There's data on Alzheimer's and other conditions that profit from dynamic analyses. So in the end, what we really want to know is how this dynamics is affecting behavior, right? So we need to at some point move on from resting state to the task setting, because we're all ultimately interested in how the brain is generating cognition, right? So although studying connectivity at resting state is extremely informative, how do we move on now? How do we understand the functional relevance of these dynamic fluctuations? So this is one example from Danny Bassett's work. And this is during a simple motor sequence learning. The participants were actually scanned on 3 sessions over 3 separate days. And they're looking at a measure that they dub flexibility, which is basically how often a given node changes its modular assignment over time. So is it always falling, if you're applying, if you're inferring the modular structure, does that node always fall within the same node, or is that modular structure really dynamic over time? So what's interesting here is in their dynamic analysis, they show that this measure of flexibility increases from session 1 to session 2, and it significantly decreases from session 2 to session 3. What's actually the most important part is that the flexibility in session 1 here in the black dots is predicting how well the participants are learning in the next session. And the same holds true from session 2 to session 3. So it doesn't predict how well you're doing in the current session. How flexible your brain assignment is and how much it's actually changing is predictive of your future learning behavior. So in this case, this is just a good illustration of how you can apply functional connectivity and in particular graph methods directly to task data the exact same way as you would do it to resting state data. But this brings about an important question. So we don't know how much of the functional connectivity during task is actually comparable directly to intrinsic connectivity in the sense of just spontaneous co-fluctuation. So you could imagine that the task is activating region A, B, and C. And so if you repeat that trial 100 times, we see a correlation between regions A, B, and C. And is that really a measure of how they are necessarily communicating with each other, how they are connected or not? Is the correlation just evoked by their co-response to the task? So that's kind of an open question. I think you discussed it a little bit in the DCM session of situations where that may be the case and that may not be the case, but I think it's an important question to think about. In any case, we had this this approach of treating the evoked responses as something other than intrinsic activity. So let's assume we can, for now, let's treat them as being something different. Who knows if that's the right approach? Probably not. But just with the idea of marrying this more standard approach of running GLMs and evoked response analyses with intrinsic connectivity analyses. Let's just, for the simplicity of that, think about these things as two separate issues. What we wanted to do in this task is evoke brain responses, but put them so far apart that there is a large enough window in between the hemodynamic responses that are not affected at all by the hemodynamic response itself, so that we can really look at activity and connectivity baseline between these single trial evoked responses. So participants are lying in the scanner. They're blindfolded. It's an extremely boring task. So it runs over 2 to 3 sessions of 20 minutes each. You're not supposed to fall asleep. You're blindfolded. Obviously, you're not moving. But the task is nothing but listening for a burst. It's almost white noise, not quite, for a burst stimulus of half-second length that is played at your own personal threshold volume, which means that by definition, 50% of the time you won't even hear it. The stimuli are 20 to 40 seconds apart, but since they're not hearing half of them, for them it's 40 to 80 seconds apart. So it's it was quite a challenge to at least to some degree, make sure that the participants were not falling asleep. So we have a few behavioral analyses to, with catch trials, for example, that were quite loud, make sure they responded, and also showing that the behavior was actually binomially distributed in terms of hits and misses. They were not hearing everything and then falling asleep and nothing happened anymore. So there's some behavioral indications. We hope they were not falling asleep, but some variability in their mental state was actually what we were fishing for. So we didn't really mind if they're going a little bit in and out of vigilant states, because we wanted to see how different intrinsic states are actually interacting and influencing these evoked responses. So this is an example of, toy example of how the time course of the brain activity would look like with randomly distributed evoked responses, but of course for simplicity I'm actually not showing the intrinsic activity, which is the important part, because that would be all over the place and then you would not see the evoked responses anymore. All right, so this gave us a nice clean baseline between the evoked responses that can be assumed to be untouched by hemodynamic activity. And so this is why we scan these subjects so long, because no one ever does a task like this, right? You don't put your time courses, your trials so far apart. You basically mathematically tease them apart in your GLM, but really we wanted a clean baseline in this case. So here in red, I'm plotting the average evoked response in all the trials that the participants actually heard, and in blue, the trials that they did not hear from these auditory regions of interest. And what's interesting is that when you go back in time, before the stimulus was played, you can actually above chance predict whether or not the participant is going to hear the next sound, just by the level of spontaneous activity, the amount of spontaneous activity that was happening in the auditory cortex at that moment. But what was much more interesting for us was, of course, going beyond the auditory cortex and actually looking at these large-scale networks. How does the level of activation that fluctuates at infraslow speeds in these networks affect the evoked response? So this is where we were a little bit shocked, but let me walk you through this. So the so-called singulo-opercular network that you see here in red, the dorsal ACC, anterior insula wrapping into the operculum, and the thalamus are the major areas, but there are some more, often also called the salience network. So this network showed a similar behavior, a little bit similar at least, to the auditory cortex in terms of pre-stimulus behavior. So what we think is that this network is really strongly related to maintaining vigilance. And when activity falls below a certain level in this network, you're just less likely to perceive threshold level stimuli. To our big surprise, the dorsal attention system shows this huge effect in the other direction. So for whatever reason, this particularly auditory detection task, if you have a particularly high activation level in your visuospatial system, it seems to do harm in terms of detecting this target. And even more surprising was the default mode network, because higher activity actually predicted more success. But if you want, I can discuss our post hoc interpretations of this finding later on. But anyhow, what is really important, so note that this study was published in 2009 and I collected the data early 2007. So what I was really interested in is understanding how the interaction, that's connectivity within and across these networks, is actually impacting behavior. And back in the day, I just didn't really know how to do that. I was looking at pairwise correlations. I was looking at the correlation matrix across the regions of these particular 4 networks that I was looking at, and I just couldn't move on from there. Why? Because I was missing the methods I was describing yesterday. When you're coming across a question that requires you to characterize the global connectivity state of the brain, then just a correlation matrix or pairwise correlations is not going to get you very far. So that's where I came across the graph theory applications that were running in my then postdoc lab, and that's what I applied. So this is a more recent study, finally, that I learned the methods a little bit. So in gray, I'm actually showing you the time window that we ended up using. So we threw away all the time windows that would have been enough to look at baseline activation levels, but not long enough to actually calculate connectivity. So we're taking really the longest intrinsic connectivity states that are not affected by the evoked response. So we throw away everything else. So you look at this correlation matrix derived from, on average, 13 volumes in my case. So some of them were longer, some of them were as short as 10 volumes. And then you go to the next window and calculate that correlation matrix again. And you can see that it's a little bit different. It's reflecting the same architecture overall, but it's a little bit different. So if you do that for every window, are you able to predict behavior just based on these correlation matrices, right? So what I did is just pattern recognition. I don't know, you guys talked about machine learning a little bit already, or multivariate analyses. So this is just straight, simple pattern recognition where I'm feeding in correlation patterns as my features. So I took the most discriminative features from this this correlation matrix, basically. Since I have so little data, and that's a result of the fact that I have so long intertrial intervals, I don't have enough data to do this within participants. So I'm actually leaving out one trial and predicting across the full group. So it's kind of a little bit of an unusual approach. But in any case, it's surprising that it actually works, because variability of connectivity across participants is much, much larger than across brain states. And I'm trying to decode brain states from each other, right? The brain state that will lead eventually to hearing the sound versus the brain state that leads to the miss. So this line represents the performance of the classifier for the real data. And then this is the permutation, I think 2,000 permutations of the labels, basically, the labels hits or misses. and the performance is significantly above chance. So you can, to some degree, not great, but above chance, predict just based on single window correlation methods, matrices across participants, their next perception. Of course, based on the data that I already showed you, if you did the same with just looking at activity level, if you pull this over all the regions of the networks that I showed you, you will also perform above chance. But what's more interesting is if you pull features from both sets, both activity and functional connectivity, you perform a whole lot better than either set alone, which we took as an indication that the connectivity matrix is informing over and above just the trivial combination of fluctuations itself. So the intrinsic fluctuations of the nodes are giving additional information, and they're different from looking at the cross communication between the nodes. But so now comes the graph theory part. So this is what I was more or less stuck with in 2007, which is I just looked at the difference of the correlation matrices. Now what I did here is I concatenated all the time windows that were preceding hits and made one correlation matrix from that for each participant. And then I did that again for I concatenated all the windows that are leading to the other percept. So for each participant, I have two correlation matrices, and this is the T-map of the difference of the two across the participants. So it's kind of informative to some degree. You see that within the auditory cortex, there's nothing happening really, but between the auditory cortex and this network for alertness or vigilance, there is an increase, or there is stronger correlation between prior to the states that prior to hits than the states prior to misses. So the difference is not completely random all over the place. Within the default mode network, there seems to be an increase. The default mode network decouples from the dorsal attention system, it couples with the frontal parietal system, and so on and so forth. You could write a whole paper just describing this, but it's just very, very descriptive. Also, of course, none of this would completely survive multiple comparison corrections unless I actually average these values within networks or within pairs of networks, right? So then it's statistically significant, but if I were to correct for all the connections, that would be very weird. So I find myself here in a situation where I really think, okay, I want to describe the overall brain state that is preceding hits compared to that precedes misses. So I moved on to a much denser correlation matrix in this case. This is actually from resting state, but in the same participants, but it's just making the same point. Here I moved to the Power and Peterson atlas with 160 nodes. I was left with 140 nodes or so. Some of my slices were missing. But the point I'm trying to make here is we had the discussion of how many nodes we need and so on. You would not be able to really derive a graph from the previous correlation matrix that I showed you. Oh, this was, okay. Very good point. I completely missed that. This is just seed-based connectivity maps from the participants themselves, individual for each one of them. Yeah. So this wasn't really an atlas. Let me show you the regions. It's so this is basically the regions. Yeah, it's of course only including four large-scale networks, and the auditory cortex is really not much. The reason is this is coming from the old paper before I was actually in this world of whole brain connectivity. I was very focused on networks that I had cognitive hypotheses about, which include the major task-positive networks, and then the default mode network, just because it's playing often the opposite role in cognition. But yeah, so these regions are basically, I was putting a seed, for example, in the posterior cingulate cortex, and then just running a GLM, extracting. Did Dr. Snyder talk about seed-based connectivity maps? You guys all know what that is, more or less, seed-based. Yeah, so this is resting state seed-based connectivity. Resting state from the same participants. I'm defining those nodes, and that's all. This would not be enough for a connectivity analysis, right? Because with the relatively small number of edges, I would have to go pretty low in threshold to get enough edges to actually construct a graph, and then at that threshold, the edges are not really meaningful anymore. So yeah, we talked a little bit about the dependence of your threshold on the number of nodes, but it's of course extremely dependent also on the quality of your edges. So when you're going to shorter windows, you have to ask yourself and how far you're trusting your correlation value, right? I have seen windows as short as 8 volumes in the literature, but I really strongly suggest to actually look and make sure that the correlations that you may derive from short window analyses, that those actually make sense. Look at the graphs that come out, see if the networks, the modules come out as you expect them, for example, right? All right, so I'm moving to about 140 regions. And then so this is from concatenated data. This is not, I'm not doing graph theory on the single window correlation matrices. I didn't trust them enough to do that. That might be possible. People actually do sliding window graph theory. So again, that depends on the quality of your data and how short do you want to push those windows. Yeah, the one example that I showed was Danny Bassett's work, right? The windows were still pretty long in that one. But in this case, I concatenated data for the pre-hits situation and the pre-misses, and I'm generating one graph for each participant and each mental condition, basically, right? So the perceptually efficient state and the perceptually not so efficient state. So that leaves me with 2 graphs, and then I can, of course, calculate the graph metrics for each of these 2 conditions over the participants and run the group statistics. So here I'm showing you one of the best case participants. This is one of the nice visualization tools. So the metric I decided to use was modularity for several reasons. One was, as I showed you, there were several studies that actually showed that one of the major characteristics changing dynamically over time is modularity. The other reason was I'm looking at this correlation structure that I have, the different difference in the correlation matrices. And they don't look random. It looks like something within certain modules is changing and something across certain modules is changing. So the modular arrangement of the brain seemed to be changing. That was one major reason that drove me to actually look at modularity. And so this is the only measure I looked at. And I found that modularity before hits was significantly stronger than before misses. So the brain is actually in a more segregated state. The single modules are better defined, better encapsulated before hits. So for this participant, if you use a force field atlas, you can actually visually see this effect. So if your difference in modularity is strong enough, you will be, with certain tools, actually able to visually appreciate it. And I really urge you to try and understand, if you see an effect, what is driving that effect. It's really good to go, for example, to visualizations and actually see what's going on. So the force field atlas, just a short moment on that, is basically assuming that the nodes are magnets that repel each other, but if an edge exists between a set of nodes, it's pulling them together like a spring. And so these are 2 opposing forces, and then they find an equilibrium between them, and that's what you basically visualize. So that's how I understand the algorithm, intuitively speaking. But yeah, so here before misses, there seems to be a lot more integration going on, if you will. There is much less segregation. So if you quantify that statistically, you indeed see across participants that before misses, the modular structure is very strongly, or I should rather say very consistently weakened as compared to the situation before hits and as compared to the pure resting state in the same participants. So this part I'm just showing to explain my approach, that I had a difference in modularity and I didn't really know what's driving it, right? So you can always go back to your measure, in this case modularity, and say, okay, well, mathematically speaking, what is it? So modularity is averaging the fraction of nodes falling within a module across to what you would expect in a random network to fall within that module. But then it's averaging that across all modules of your system. So in this case, it means that there might be different contributions from the different modules to this overall measure, because it's an average across all of those. And that's why I broke it down. And I found that the most consistent reduction in modularity or reduction in contribution to modularity was actually observed in the default mode network and in the visual networks. So one more step that I took from there is the idea that modularity is looking at how much clustering is happening within a module as compared to cross-communication that is integrating across modules, right? So my next step was to actually look at those two aspects separately using nodal measures. So this is basically just a follow-up to further understand why I was seeing this effect. So on the left-hand side, you see within module degree, which is a measure of clustering that we discussed yesterday. And then on the right-hand side, you see participation coefficient that is actually measuring how much a given node is actually communicating with a diverse large set of other modules. So I plot these for default mode in visual networks just because I saw the strongest modularity effect there. And it turns out that both the within module connectivity has changed as well as the participation with other modules. So this modularity effect is driven by a change in both quantities that indirectly contribute to modular assignment. And then another thing that I think is nice if you're looking at nodal measures is actually plotting them on the cortical surface. This gives you an idea of how they're distributed. For example, the participation coefficient in my case seems to be fairly distributed across the cortex. So most of the nodes are showing a increase in participation coefficient before misses. But the within module degree was much more restricted to certain areas, for example, here to the frontomedial node of the default mode network. All right, so that's, I think also just visualizing your data in various ways can help you to actually understand where your effects are coming from, or why you don't have any effects. It's also possible, right? So the question was if I would have expected something in the auditory network. It's a very good point. Yeah, especially because I actually saw the connectivity change between auditory and singular opercular in the correlation contrast matrix. But it seems to be so selective to certain networks that it may not have come through. Are you asking if I would have expected something here in terms of modularity contribution? Not necessarily. So in terms of within module degree, the auditory cortex internal correlation did not seem to change actually very much, right? If you think about that contrast matrix at the coarse level, this one here, well, okay, in this case I have only 2 auditory nodes. But if you were to look at the high resolution version of this matrix, excuse me, there's not much happening in between auditory nodes. So within module degree, I wouldn't expect to change. But you're right that I found if averaging this whole block of connectivity between auditory cortex and this vigilance system, that there would be an increased participation, if you will, and a decoupling from the default mode network. I don't see that maybe because this is so selective for these 2 networks. So again, the participation coefficient is averaging it's basically measuring how diversely you're connecting across the board with the other modules. So it's a very good question you're asking actually, because maybe I should have added that analysis to it. The only thing I did is really presenting the average of this, which answers that to some degree, but I didn't look at the whole brain level. And so you're right. If you were to see my results, the next obvious question would be, okay, so why don't I see something in the auditory cortex? Does it mean that there is only these changes and it doesn't really change very much overall? So then you could find other nodal measures and maybe more specifically look into that question. So one answer might be that in this case it's going into opposite directions, so this would completely cancel out in participation coefficient, right? So the communication to this task-positive network is increasing, but the communication between auditory cortex to the default mode network is actually decreasing. So these would completely cancel out in all the measures that I actually looked at, yeah. And yeah, in terms of clustering, no, there is not increased clustering. Yeah, good point. But yeah, I think the one strength of this is actually showing that whether or not you hear a threshold level sound, even this very, very simple task is actually determined by your entire global system connectivity state. It is not just the auditory cortex. So, I'm closing with these last few slides. I think I'm running a little bit late. This is basically an image of energy consumption in the body at resting state. And what is astonishing is you look at the heart, which is a muscle that is beating all the time and is consuming so much energy, it's so active, but it's nothing compared to the brain. And this is not even when we're cognitively active. So now I was saying that you can look at the raw bold signal from your task. This is the same task I just showed you. This is the raw signal from the auditory cortex. This is 3 of those randomly distributed evoked responses, and then look at the background activity. If I didn't show you where these are, you would barely recognize them, right? So now you could say, okay, bold activity, there's a lot of physiological and non-physiological noise in there, so how do you know this is actually neural activity? How do you know this is related to this enormous amount of activity that is going on in the brain in the absence of task? So I looked at this question using electrocorticography, and this is one electrode that was highly responsive to an auditory sound. And the reason I know that it's highly responsive is that you see very strong high gamma power just after the sound was played. And this is the average evoked potential at this electrode in response to the sound. So now the reason we're averaging so much in neuroscience is that this is the raw data. This is pure electrocorticography. You won't get any closer to the human brain than this, right? So I played the sound here, I think 6 times. And if you were trying to find when it's happened, it would probably be a little hard to be sure, right? So these are where the sounds happened, and some of the evoked responses are pretty clear, but some of them look nothing more than what's happening in the background, right? So what's going on here? Very, very interesting, I think, if you're looking at raw data. And of course, animal electrophysiological studies are going to confirm this, have confirmed this already, right? So most of brain activity, and I think Brakel's group is estimating something over 85% of brain activity has nothing to do with what is going on in the outside world. And we are almost never studying those 95%, unless we happen to do intrinsic connectivity analyses, for example, various resting state analyses that are becoming more prevalent. So, but most of our approaches completely ignore the largest part of the brain's processing architecture, and we're still trying to relate brain to behavior, which is astonishing because we're leaving out really this big part. So this picture is basically the final point I want to make is that at different scales of space and time, there is an intrinsic architecture that is replaying and displaying exactly what the co-evoked activity in the brain would look like. And this holds true both at this level, and Camille was talking about a lot about these pinwheel orientation columns, basically, so I don't have to say much about them. But with optical imaging, you see this spatial resolution at a millisecond level, and these maps, they exist even in the absence of vision. This is an anesthetized cat, right? Completely absence of vision. And in this case, they're iteratively flipping through these iso-orientation columns. So there's activity in the neurons that are particularly responsive to vertical directions, and then 40 milliseconds later, it flips over to another orientation and back and forth. And then you go to higher levels. So in this case, this is from John Dylan Haynes' lab, where they used, this is resting state again, fMRI resting state in a conscious human. They used a seed in V3, and they are looking at where the spontaneous fluctuations of activity in V1 is predicting the spontaneous fluctuations in V3. And it happens to be very, very focally in the region that in retinotopic space directly corresponds to this region in space. And then you move from there to these larger scale networks. So for example, in the visual cortex at resting state, you see fluctuations that can separate central and peripheral vision. And then you can zoom out one more level. Now the visual cortex is all just one big module, and the other modules are also at a coarser level of hierarchy. So the point I'm trying to make here is that the brain seems to constantly spontaneously flip through architectures or connectivity states that each one is good for a certain kind of calculation or a certain kind of cognitive process, certain kind of perception. And the brain is flipping through these in an iterative way. And what we're suggesting is that that may not only be the case because the brain needs to maintain the synaptic connections between the functionally related regions, for example, between all neurons that are responding to a certain orientation, but it's also because the brain is dealing with an incomplete sensory world. So we have to constantly predict the world. The brain is constantly anticipating what it would be perceiving. The brain is constantly anticipating cognitive challenges that may arise. And it seems that the brain can't really be at this on state in all of its functional architectures at the same time. So it's maybe iterating through these states in order to check them one by one, give a chance to each one of these cognitive architectures. So that's a little bit of hand waving, but there is some data to support that, and we've discussed it in this opinion paper if you're interested in going into that. So I'm just going to close with this almost last slide. This is data that is directly comparing cross-correlation matrix in a large number of participants during resting state and concatenated data across a wide number of tasks. And they are extremely similar. And if you're looking at the modular partition derived from this correlation matrix versus this correlation matrix, they're also highly overlapping. So the question then becomes, is it actually a good idea to think about evoked responses as something so different from these intrinsic fluctuations? Maybe not. Maybe what's actually happening is we're thinking about the brain as some sort of a still background where nothing is happening in the background, or it's uncorrelated noise, or it's some kind of white noise that is not affecting cognition, and then comes in some sort of a stimulus into the brain. It elicits this wave, this brain response. And if we do this often enough, then we can see the average brain response and understand how cognition works, how the brain is solving or processing this particular stimulus. But looking at the vast amount of brain activity, we actually know that the situation looks like something like this. So the incoming drop of water, the wave that it's creating is interacting with all of these other things that is already happening in the brain, and they are interacting with each other. And actually, the waves that are already existing in the brain are the vast majority of what is happening in the brain, not the evoked responses. So the processing of information not only depends on the initial brain state, but it may actually only cause a subtle deviation of the iterative dynamics of function connectivity in the brain. So let's say you were at resting states, right? Your brain is going through these different kind of if you will, connectivity architectures. Maybe something arises and your brain is shifting into a slightly different connectivity state due to that. And that may be all that's happening with evoked brain responses. So really cognition is an interaction of what your brain is doing with the outside world. So we all know that we're predicting a reality that is not really corresponding to anything out in the physical world. And that's reflected in how the brain works. So I think I opened probably more questions that I answered, but I'm summarizing those probably somewhat hand-wavy suggestions here. And yeah, thanks for listening a little extra long. Okay.
So I have a question. It might look a bit silly, but we are assuming that at the time brain is in one state. However, it might not be the case. Might be that some regions of the brain are in a different state, and the brain has one dominated state. And this seems more plausible to me, given that when we are going like from one state to another state, there should be some like non-dominated state in the brain within some regions there that is predicting the next state of the brain. And that is how it is like transferred maybe to the next state. It's just the way that I can think of. I don't know.
I'm not sure keep the microphone because I'm not sure I completely understood. So you're thinking the brain is mostly in one particular state?
During one particular task that we are doing, or during all the analysis that you did, we are assuming that the brain is going from one state to another. So at the time, brain is only in one state, right? What I'm saying is that it might not be the case. I mean, at one time, the brain might be actually in, like, it might have several states in it, like inherently, but one of them is dominated.
Okay, but then the combination of those states, let's say it's a combination of 3 states that are somehow, let's say, linearly overlapping with each other. They would together, if you were looking at the entire system as a whole, together again form one state, right? One aggregate state.
Yeah, so what I want to say is that why are we not looking within those like sub-states, let's call them? That might also be interesting, to look at those sub-states of the brain, see how are they different, or how many sub-states could exist.
It's a very, very good question. Right. Very good question. Because in spite of the studies that I showed you that were so beautifully also using different numbers of clusters in that k-means study, for example, the results seem to be converging, but it doesn't quite, with 100% certainty, respond to the question of whether we're actually in a multi-stable state where we have a discrete set of states and we're iterating through those, or it's an infinite number of states, right, that we're going through. I think we don't have an answer to that. Although both the simulation studies and the dynamic functional connectivity human studies suggest that there are states that are to some large degree similar to each other, enough so that we can group them by their spatial distribution similarity. Yeah.
I like this idea of looking at connectivity during tasks and hits and misses and things like that. I guess the only problem I'm having with it is that I have a hard time, and you find this difference in modularity or something in the default mode network, but what does that mean in terms of what's going on in the task? I mean, people can look at detection tasks or discrimination tasks and look at early sensory areas, and they can look at noise in terms of hits and misses, and look at signal that's not correlated with the stimulus, but it is correlated with the ultimate behavior, whether you hit or whether you detect or fail to detect, or what your performance is. And there, this correlation with noise makes sense because there's a clear link to the task performance, and it's in the area that you know is underlying the task. The thing I find hard here is, like, you know, this change in modularity was in a visual network and was in the default mode network. I don't know if there's a way to go back and say, well, is it that the default mode that correlation across the default mode network is increased or decreased before hits or misses? I guess I'm just trying to tie it back to sort of like, how do we think the actual physiology is working?
Absolutely.
I understand looking at connections and distances between them and things like that in this very abstract space, but how do we bring it back to?
I completely agree that we should really try to tie it back to how we think it may actually relate to cognition, because then I'm looking at the entire system. Why would that matter for a single, white noise that's happening? Why would that? So one reason I split the contribution to modularity into the subsystems was I was wondering where it was coming from, and the visual system is not important for this task. As I said, they were also blindfolded, and in fact, looking at the activity level of the visual spatial system, it looked like higher activity was actually, deteriorating performance, right? So my interpretation, post-hoc interpretation of this is that encapsulation of the default mode network and the visual network, both of which are not important for this particular task, is beneficial for perception. And that's why the strongest within module clustering, the strongest difference in clustering is occurring in those two systems as compared to the other systems. So that's one point. And then in terms of the correlations, yes, correlations were stronger within the default mode network prior to hits. That's what I showed. It's true that it's hard to connect the entire system's situation to that task, but I think ultimately we have to. So where I started off with was a correlation matrix, this coarse one, for a set of networks where I could easily interpret hand wavy as I just did, why increase or decrease in clustering or correlation or whatever may be important for this task. From that, I moved on to the question of, okay, we are a complete system. When we're performing any task or we're reacting to anything in the world, the situation in the entire system matters, not only in that isolated system. And this comes back to a little bit to the answer I gave to the question yesterday, where I was asked whether I can think of situations where a particularly highly segregated brain state might be better than an integrated brain state. And this is an example of where an overall segregated brain state might be beneficial. And I'm pretty sure that you will, if you repeated the same experiment with like far apart trials, but highly kind of tasks that require a high back and forth of information rather than just simply saying, oh, I have enough evidence for a beep, so I click. So something more engaging, then you may see the opposite effect. In fact, I'm predicting you have to, because otherwise it'd be weird that we evolutionarily evolved into brains that go back and forth between high and low segregation, right? Some of it will be good for certain kind of cognition, some of it will be good for certain other kind of cognition. And we're going back and forth at different spatial and different temporal scales. That's what I'm thinking the brain is doing all the time. So in the end, you have to move on to connect the entire brain state to how it may affect perception, even though I'm agreeing with you, we're getting pretty abstract there.
Yeah. Do you have enough data to look at the false alarms, if there are any?
Yeah, really good question. Let me actually try to find that paper. So Carl Friston got his hands on this data. Guess what? He's like, oh my God, predictive coding. Okay, so we had enough. We had enough data to look at false alarms before in the activation. There was a clean enough baseline to just look at 2 or 3 volumes of the baseline after the previous HRF had ended, but I didn't have enough to actually calculate correlations. So unfortunately, I cannot look at connectivity states, how they influence false alarms, but we analyzed this. Oh, this is actually the wrong experiment. We analyzed this for the false alarms in the auditory cortex, and I mean, I was just shocked, and Carla's like, okay, of course. So yeah, this is Andreas Kleinschmidt's work mostly, and his really thinking that comes from this London school, I think. So what he's thinking is happening is that, as far as I can recapitulate, is that if the activity in the auditory cortex would go particularly high, you would say, oh, maybe that was a sound, so let me click. But the predictive coding notion has an additional mathematical term for the precision or the reliability of your evidence. And so for some reason, sometimes the activity not only goes low and you just miss the sound, but it actually falls so far low that you can no longer negate the absence of evidence. So the reliability of your signal from this region is so low that your top-down prediction can no longer be negated, and then you elicit a false alarm. Again, this was way beyond me when this paper came out, but what is astonishing is that we, so this is Guido Hesselmann's work, we saw the exact same situation looking at an entirely different task. Again, far enough apart stimuli, again, threshold stimuli, but in this case, it's coherent motion detection. So the motion coherence was so low that on half of the trials, participants said, oh yeah, this was coherent. On the other half, they said, no, I don't think this was coherent. And again, if you split the behavior by correct and incorrect, in this case, you have 4 responses, then you see a very similar situation that is actually in line with that model. Yeah, unfortunately, yeah, we don't have enough false alarms to look at that connectivity. That would have been great. I should probably go back and look at the concatenated data. The problem really is that subjects don't have any false alarms at all. They may have 1 in 20 minutes, you know. You cannot do any group statistics. I could have some descriptive visual presentation for those that do have a whole lot of false alarms, but it'd be hard.